Good morning to all. Once again, a warm welcome to this workshop on advanced drug design and medicinal chemistry 2022. We are lucky to have with us Professor GPS Raghav from Triple IT Delhi. He has done PhD from IMTech Chandigarh and postdoctoral fellow at Oxford University, UK. He is a bioinformatics specialist with vast 30 years of experience at IMTech. He has won numerous awards. He is a fellow of National Academy of Sciences and the Indian Academy of Science, Bangalore. His research interests are bioinformatics, applications of protein modeling and engineering, genomics and informatics solutions for integrating biology, integrative approach for designing molecules for cancer therapy, and applications of bioinformatics in system biology. He has also he is also a great teacher of applications of artificial intelligence in biomedical sciences and big data mining in healthcare. Today, he will talk about advances in computer-aided protein therapeutics. With this brief introduction, I welcome you, sir, and over to you, sir. Thank you.
And thank you very much for nice introduction, and thank you very much for giving me opportunity to present the work. So today, as you can see the topic, advances in computer-aided protein therapeutics. Why I am giving a lecture on this topic? Because this is my favorite topic, and nowadays, a lot of drugs have been approved which are based on the biological molecules. In biological molecules itself with the protein therapeutics basically. Protein or peptide therapeutics basically. So peptide is nothing, it's basically a small size of the protein, we call peptide. So I will use this word exchangeably. Sometimes I will call peptide, sometimes I will call protein, but they are the same thing actually. Okay. So if you are interested in this area, then you will have my website. You can access my website, and on this website, large number of web servers are available, and that include servers, softwares, which are useful for protein-based therapeutics, basically. I know this conference is mainly on the drug discovery. So recently we write a review. I will present few slides on this review, which will be useful for the participants. And that you can see the title is too broad. Computational resources in healthcare. Healthcare is wide word. It includes all your chemoinformatics, pharmacoinformatics, genomics, proteomics. Because if you see carefully, most of us are working to provide the better health. Whether we are in the life sciences, whether we are in the chemistry or we are in the pharmaceuticals, most of the researchers' aim is to provide the quality of life. Okay, so question is, can we have some review or some source from where you can get all the resources? I'm talking about computational resources, from where you can get the complete and comprehensive information. And when I say computational resources, it includes database, repositories, include web servers, prediction models, standalone softwares, mobile apps, or any other resources which directly, indirectly help you in the healthcare. So this is a recent review, and it is available. You can have a look on it. Because it was not practically possible to cover all the resources, computational resources in a review. So we also have the website, and this is called Portal for Health Informatics, and this is the address of website. And this website have the all type of computational resources organized in the form of genomics, proteomics, drug informatics, medical informatics. It means we have organized different resources in the different subject areas, so you can easily cover them. This is the one page I have shown you where we have clicked on the medical informatics. For medical informatics, again, it is in the subtopics basically. Under the medical informatics topic, we have the subtopics like personalized medicine, disease biomarkers, clinical informatics, disease forecasting. If you click further on, let's say, clinical informatics, then all the resources related to the clinical processing, data processing, you will get these resources. So we have tried our best to collect and compile as many resources as possible and put in this website. So if you are really interested in computational work, then I will advise just to visit this website. You will get most of the information from here. Okay. After this regarding the review, because health, when we say computer research for healthcare, then what we realize actually, we read the literature, and what we realize actually that they are available in different form. Means some people are writing the review where they are saying that application of healthcare in application of biotics in healthcare. Some review are there, application of chemoinformatics in healthcare. Some is on the pharmacoinformatics, clinical informatics. Even some review are there on the AI in healthcare. So what we thought that take all the fields, compile all the fields, and put it to the way where the simple person can understand what type of resources are there and how we can achieve them. So we combine all these fields. After combining all these fields, we ultimately, because they are all connected with the healthcare, we ultimately divide our review into the 5 parts. One is for the drug discovery, and drug discovery may be peptide-based or protein-based or maybe small chemical. Because if you see carefully, by traditionally, most of the drug discovery is focused on the small chemicals. So we will focus on the small chemicals based, and later on I will talk on the peptide based. Okay. Then one of the problem you face or anybody face in the field of drug discovery is toxicity or adverse effects, or you call it simply ADMET. Okay. So the different side effects of the drug discovery. So I will cover that. What are the major tools actually? Then next part is basically again peptide and protein based. That's basically vaccine development. As you know, that in case of the coronavirus, one thing which saved the population heavily is due to the vaccine actually, not due to drugs basically. So we talk about the two concepts called innate immunity system and adaptive immune system. Innate immunity system is important for designing the vaccine adjuvants or activating your immune system. And adaptive immune system is required to generate the memory cell or generate the antibodies which are specific against the particular virus. So I will cover this one when I will talk about advances in protein-based therapeutics. Basically. Another important part is disease biomarkers. Because nowadays, due to the advancement in the genomics, proteomics, and other fields, there is a number of disease markers can be designed, which are basically predictive biomarkers, prognostic biomarkers, where we want to understand the life expectancy of the patient, basically, or image-based biomarkers. Okay. In addition to these biological means, there is another area which is coming up very fast actually. That is basically IoT, Internet of the Things actually. IoT is coming up very fast actually. Because remember one thing, if you are a researcher, you cannot avoid the recent development in the field. One of the problem in traditional researchers in India, or not in India, everywhere actually, they feel they will be better if they focus on only in one small area actually. But time is changing very fast. And if you are in the field of informatics, any informatics, bioinformatics, chemoinformatics, doesn't matter. you will not be able to survive if you are focused only on a particular small area, because your dependency is on the data. And if you will not get the sufficient data, you will not be able to do much actually. So it's used to be little bit opportunistic in nature, and you have to see what type of data is generated in the literature, and then accordingly you have to modify yourself actually. So IoT is another field which is very coming very fast, where there are number of mobile applications. For example, smartwatch, where you get the information about your heart rate and how many steps you have moved. This is just a small example. But there are a number of mobile apps where they can check your medical conditions, basically, including the Parkinson disease, the severity of the Parkinson disease, blah, blah, blah. There is another area which is very popular nowadays, is telemedicine, actually, through the telephone or even through the medicals. Let's say in last 2 years, we suffer heavily from this COVID-19 pandemic. How we survive through the online. There is a lot of medical consultancy through the online, Google Meet or another platform actually. So these are the interesting fields are coming up basically. So telemedicine is very important. Sensor-based technology is coming up very fast, where the sensors are become much cheaper and they provide you health-related information. So in this topic, I will cover what type of major tools are available. It is not feasible to cover all the tools and techniques here in a review. I will show only the major tools, softwares basically, or resources, computational resources, not all. If you are interested to be a little more, you have to visit our website actually, where we provide the more information about this part. So let's say I'm giving you the example that small chemical-based drug discovery. If you are working in this area, then what tools are available? Why I am working so hard from last number of years to collect and compile the resources? Because I feel the countries like India or developing countries, it's really impossible to afford the commercial softwares. Frankly speaking, I'm not against the commercial softwares, but one side we say that India doesn't have the money or grants, and other side we are wasting our money on the commercial softwares, and these commercial software are black box basically. So you are not improving your science unnecessarily. You are using some softwares and then just you are paying money actually. Please remember one thing. Nowadays, open source software or free softwares are as strong as any commercial software. And they are freely available. You can modify them. You can use them actually. Okay. So I will advise you, particularly young generation, because old generation, it's really difficult to change actually. Except few persons in the old generation, it's difficult to change. They will do whatever they are doing from the last number of years. But for young generation, I will advise you work on the open source platforms or free platform, where in the science you help each other and grow basically. Okay, because the ultimate goal should be for a scientist should be to bring the medicines at the affordable price. If you are not able to bring the at affordable price, a lot of people will die without the medicines basically. So anyway, I'm giving you some major resources. Let's say if you are really interested in the developing the different type of chemoinformatics tools actually, or pharmacoinformatics, the PubChem is the best database or resource where large number of assays are freely available. You can use them, you can develop the chemoinformatics tools for your work. And it is freely available. So this is a major source for anybody who's working in the field of chemoinformatics basically, or pharmacoinformatics. Same way, there is another resource, CHEMBL, database of drug-like molecules. That's another database basically. So I'm not going into all. These are some of the databases already available to the market. After the databases, these are some of the databases. Then there is a question when you want to develop any prediction method. Because even if you use the common sense software or freeware, the major thing is structure descriptors. Because QSAR, what is a QSAR? Quantitative Structure Activity Relationship. Okay. So in QSAR, what you need? Descriptors. Because when you talk about machine learning techniques in this area, you are basically talking the classification of different chemicals, small chemicals. So when you want to, let's say, apply the machine learning technique or any technique, you need to calculate the descriptors. Descriptor, or maybe you can call it feature, or maybe you can say characteristics. These different words are there, or attributes. It means you have a chemical, you have to calculate the different property of this chemical so that you can discriminate the different chemicals based on their properties. And the properties are represented by descriptors, fingerprints, characteristics, whatever you call it actually. But please try to understand the fundamentals because we want to understand the properties of the chemicals, and that can be presented in the form of descriptors. One of the powerful software which is heavily used is Pedal, which is based on the original software CDK, but it provides you large number of descriptors. There are other descriptor softwares are there, Moderate and other descriptors basically. So these are the free softwares which you can use for your work day to day. Actually, you don't need to pay any money to anybody. Besides that, there are a number of softwares which are for the docking. You know, the docking is very important when you know the structure of the receptor basically. So these are some of the tools which you can use. And I will tell you the number of benchmarking I have seen actually. These freewares are as good as any commercial software. So you don't need the money. Please don't try to get more and more grant and waste on the commercial softwares. Focus on freewares. They are sufficient for your research. That will save you a lot of money and a lot of your headache basically. Then there is some other softwares Weka to implement the machine learning techniques, Tinker. So I'm not going in the each and every one in the detail because that will take a long time actually. But idea here is on this our website or in this review, you can get most of the tools and techniques which are freely available for your research if you are working in the field of drug discovery, computer-aided drug discovery. I'm talking about the small molecules. Okay. The next question is basically to predict the toxicity or maybe ADMC properties. So these are some of the tools. One of the tools we have recently developed because we are the active developers in any field of drug discovery, and this is one of the recent tools developed by our group. First time we predict the molecules which can cause the allergy. Because there are a number of tools for the predicting different type of toxicity, but there was no tool for predicting the allergy of the chemicals basically. So last year we developed this tool and we identify a large number of molecules which is approved by FDA have the allergenic property. And we checked top of them and we found that there is a literature evidence for that actually. So idea here actually that you have to use these tools for your research when you are working because it is possible that you are able to clear your drug at in vitro level, your activities, but it may fail in the clinical trials and you will lose a lot of money. So it's better to check it in advance whether your molecule is druggable or non-druggable. So this is the link for the peptide-based therapeutics. I will cover later on actually. This is for the vaccine design. I will cover these slides later on actually. This is a basically page where you will get the information related to biomarkers and particularly genomics-based biomarkers like classification of early and late stage of the cancer, database of cancer enhancers, liver biomarkers, how to identify the liver cancer, identify early stage of cancer patients. So there are a number of tools are available, and number of tools have been developed from our group and from other groups also. So this review is not for tools developed from our group, but this review is for all the tools developed in the world actually. Unfortunately, fortunately, most number of tools have been developed from our group and which has been included. It's look like that we are biased towards our tool, but this is not the fact. Okay. Same way, there is another concept nowadays that's online tools basically, which is important for mobile apps and health informatics. Like what I'm just giving some examples. One is Aragia. You know very well how this app help us to fight against the coronavirus. Just think about it. This this Aragia, it helped us a lot in whether it's a vaccine development or any informations. Play an excellent role. You cannot think without this this app, how to survive actually in the last 2 years. And there is another these are the examples actually. Another example is an online pharmacy with a wide range of prescriptions basically. Okay. Then we have the other tool, e-Sanjeevani. So here it's not all tools, but some of the representative tools are there. So the idea here is just to tell you that this field, mobile apps based healthcare, is coming up very fast actually. Okay. So you have to learn the new field as well. Because sometimes as a researcher, you have to understand each and every field got the saturation after a certain time. If you stick with that field, then you will be also saturated with the field and you will become the obsolete in the system. If you want to maintain your presence or active role in any system, you have to change with the change in the system. Because in this world, only the person will survive who change with the time, who change with the change in the system. Otherwise, you will be out of the system. So these are the fields which are coming up fast basically. Now, because the ultimate goal of today's talk is to talk a little bit more about the advances in the peptide-based or protein-based therapeutics. Frankly speaking, when I start this field, or I thought about this field, let's say 6-7 years back, I talked to the number of chemists in India. And I discussed about the new field, the protein peptide-based therapeutics, because we have the expertise in the area of bioinformatics. So we want to come into the drug discovery from the bioinformatics point of view, not from the chemoinformatics point of view. Okay. So a lot of people criticize, they say, no, because most of the chemists with whom we interact, they are traditional chemists and they have the traditional knowledge, old time knowledge, where they believe that biological molecules based therapeutics is a fancy area. It's not going to come in the future. Only area which is used practically is small molecule based drug discovery. So anyway, I talked to them, then what I did actually in our group, we go to the FDA. You know, FDA is basically Food and Drug Administration database where they maintain the number of drugs and food-related information, which is acceptable worldwide because they maintain certain standards basically. So ideally, if you want to sell your drug in the market, in the world, then it should be approved by FDA. So FDA is a major agency. So what we did, we simply collect and compile all the FDA approved drugs which are based on the peptide or proteins. And to our surprise, a large number of drugs approved in recent years they are based on the peptide and proteins rather than based on the small chemicals. As well as failure of these peptide and protein-based drugs are less in comparison to small molecule-based drugs. Okay, so that's a advantage. And this is a website. And believe me, in last 3-4 years, this website have been heavily used worldwide, and people are asking for the update basically. More than 290 citations have been already come in last 3 years. So you may understand that this site is in heavy demand. And now we are in the process of updating it to get the up-to-date information in this site. Now, just one important part is when you talk about the protein-based therapeutics, then it has to be classified so that one can understand, user can understand what type of protein or peptide-based therapeutics have been approved and used in the market. These are the 4 major groups. I'm not going in the subgroups. Group 1, when your protein in your body is defective. Let's say, for example, your insulin become defective or it has the mutations and it is not serving the purpose. What you can do? You can provide the insulin, normal insulin, from the outside. Or another possibility is there that your system is not able to make insulin in sufficient amount. You can provide the insulin for outside. So it's basically augmentation the existing proteins or existing pathways. So means your body is not able to produce the sufficient biological molecules you are providing from outside. Okay, so this is the one group of therapeutics which is heavily used in the market. Another type of application of protein and peptide is drug delivery, because protein and peptide is much bigger than the small molecules, and these protein and peptide have the properties, certain properties, which can be utilized to deliver the small chemicals on the right target. Let's say, for example, you have some drug, small molecule drug, it becomes drug resistant to some bacteria because membrane of the bacteria become rigid and it is not allowing to enter the drug inside the cells. Then there are some peptide and protein which can penetrate these membranes. And if you attach the drug with these peptide, they will deliver the drug inside the cell. So this is another area, drug delivery of small molecules through the protein and peptide is coming up very fast, basically. Okay. Then group 3rd is, you all know about it, vaccines. You know, when coronavirus come or any pandemic come, drug is not the solution to stop or control any pandemic. Best solution is to provide the vaccines. And you can see in the coronavirus case that we are able to save a lot of life due to the vaccines. Otherwise, this omega wave can destroy anything actually. Due to the vaccines, we got a lot of protection actually. So this is another category where we can use the peptide and protein for designing the vaccines. And group 4 is basically diagnostic agents basically. It has been shown in body fluids in certain disease that certain molecules are abundant, means they are found in more amount. Based on that amount in the body fluids, you can diagnostic a particular disease actually. So disease diagnostic is another important area. Now, before I go for the vaccine, I have to just say few words about the vaccines. If you go to the traditionally, we know the vaccine concept start from the long time back when we are suffering from the smallpox or chickenpox. At that time, it has been observed once you are infected by smallpox, then and if you survive, then whole life you are protected. It means there is something memory in our body, or immune system have the generated memory which recognize in the future and remove it. Based on the concept have come of the vaccine. Normally, if you go to the traditional vaccines, they take the virus. Let's say this may be virus or bacteria. They kill the organism or initiate it so that it cannot replicate and give to the body. And I can give you the good example. Till today, this concept is used. For example, Covaxin, which we are getting from the Bharat Biotech, is nothing. It's based on the traditional concept of vaccine, where they have make the coronavirus non-functional and give to your body. Body thought that enemy is there, and they will generate the memory cells which can fight against this particular virus, coronavirus, as well as it will generate large number of memories which will be stay in your body. So next time when you are really infected by the coronavirus, then your system already have the memory cells, B cells, T cells, as well as antibodies. So they will fight very fast and they will remove from your body. Actually, that's a concept. So still it is used. People thought, why we should give the whole organism? Can we give only one protein? Yes, that's called recombinant vaccines based on the purified antigens. For example, the other vaccines like Covishield, Moderna, Pfizer, they are based on only one protein, and that is called. So on the surface of the coronavirus, there is a spike proteins. There is a spike protein. So they have, instead of giving the whole virus, they have given extract only this protein, because nowadays synthesizing a protein is not a big deal about it. So they synthesize this protein and give to your body, and body will thought that enemy is there, and they will generate the antibodies and memory cell against the spike protein. So when next time you are infected, then you already have the antibodies which will fight, which will bound with the spike protein, which is surface protein of coronavirus, and will destroy this virus actually. Okay, that is quite heavily used. And still the work is going on the epitope-based vaccines, where they are taking the small peptide like 9 amino acids or so. That type of work, a lot of prediction methods have been developed. So I'm jumping due to the time. So if you are really interested in the vaccine development, you have to understand the whole system. How the disease-causing agent, which may be bacteria, fungus, parasite, virus, they may attack, how the innate immune system react, how the adaptive immune system react, and how the memory cells generated, how the different type of responses come. Based on that, you can design the vaccines in the future. I'm not going in the detail because that is not practically possible for me, and I'm just giving some tools basically. Like one of the example I will tell you is, in case of the coronavirus, number of deaths is due to the overexpression of IL-6. IL-6 is an interleukin. When COVID-19 infect the human, in order to contain it, in order to fight, IL-6 has to be produced actually. But in this case, IL-6 have become too high actually. Its overexpression was there. Due to this overexpression, the IL-6 increased too much in most of the COVID-19 patients, and this IL-6 injured or harmed the different tissues. So a lot of people have not died due to the coronavirus. They die due to the cytokine storm. Due to the cytokine storm, and one of the cytokine is IL-6. So it's important to understand which peptide is inducing this IL-6 so that it can be controlled. So we developed the recent method last year for the interleukin 6. These are the advanced methods. Earlier, the people are working on the MSC binders or T-cell epitope, but we especially work on the peptides that can induce a specific type of cytokines, because controlling cytokines is one of the important job in the area actually. So that's called immunotherapy actually. So immunotherapy is a direct way to control the different disease. This is this is a database because most of the people are focusing only on the adaptive immunity or B cell memory, T cell memory, antibodies. But there is another important factor, that's basically innate immune system. And most of the vaccine adjuvants, most of the adjuvants or drug adjuvants or vaccine adjuvants, they activate innate immune system. So it's important to understand the innate immune system. So this database will help, means collect and compile all the pathogen associated reception, pattern recognition receptors like toll-like receptors, as well as the ligands actually, so that you can design the better vaccine adjuvants. And this is a example. We developed the tool Vaccine Pad for predicting the peptide-based vaccine adjuvants, RNA-based vaccine adjuvants, or DNA-based vaccines. So different biological molecule can be used for the vaccine adjuvants because the chemical-based adjuvants have a lot of side effects in the long term. So this is just example that what type of tools have been developed in the past actually and what is going on. So these are the tools developed from our group. So I'm jumping on them. All are available to the public. Okay. There is an interesting part that drug delivery is one of the major applications. So there are a number of cell-penetrating peptides which have the property that they can penetrate the cell. So they can deliver the inside the cell actually. So what we did in this one, because natural peptide have a number of problems, half-life is one of the major problems. To increase the half-life, you need to do the chemical modifications. But after chemical modifications, it will remain the cell-penetrating peptide or not. That's a challenge. So this tool allows you to predict the chemically modified cell-penetrating peptides. Simply, this is a recent tool, new version for anti-cancer peptide, because NTCP we developed long time back, and we have developed recently new tool. That's an advanced tools for predicting anti-cancer peptides. Similarly, antifungal peptides. So I'm jumping. One of the major problem in literature or the peptide-based drugs is half-life. So this database maintains the half-life of the different peptides in different body fluids, basically. And there is a prediction method which can predict the half-life of your peptide. This is a new method for toxin pad. Though we developed this toxin pad few years back in 2013, but from last 2 years, it is heavily used basically. So we have come up with a new version which is more accurate than the previous version actually. Same way we have come up with an allergy actually. This is for the protein allergy actually, allergens basically. Because nowadays you are getting GMO, genetically modified foods and other things actually. So it's important that you have to predict the allergy of them actually. So I'm just jumping few slides. This is for the predicting the structures of the protein. If you are interested and you want the structure to predict of the peptide, this tool is there. It allows you all type of peptide, say cyclic peptide, modifications at N-terminal, C-terminal, natural amino acid, non-natural amino acid, all type of peptide structure it allows to predict. There are no tools which is similar in the nature like peptide structure model. So you can use them if you are interested. So this is just example of peptide-based therapeutics. I am jumping because these slides are available. I believe that they will be available to you, or you visit my website actually. Okay. So this is the example that in last 1 or 2 years since the coronavirus, our tools are heavily used because most of the tools related to the peptide therapeutics or vaccines, they are heavily used in last few years because everybody is jumping towards the protein-based therapeutics or peptide-based therapeutics rather than going for the chemical-based therapeutics. Though we work in the different areas, but our tools, which are basically protein or peptide-based, are heavily cited in last few years actually. That's so the trend is going towards a biological molecule based therapeutics. Basically. That's all for today. Thank you.
Thank you very much, sir, for a wonderful talk. I am sure that the audience has really gained a lot of information and resources and how to use these resources. We are thankful. Our own students will also be benefited by this, sir. Thank you very much, sir. Now I open this for any questions from the audience.
Hello. Uh, Thank you, sir, for your knowledgeous information. Actually, sir, I have one question regarding in designing of protein therapeutics. The major concern is the high production cost when we go for protein therapeutics designing. So, how we can reduce the high production cost for the protein therapeutics?
Frankly speaking, I have no idea about this area, that synthesis area, that how to reduce the cost. But I know one thing. Nowadays, number of companies are there on the peptide therapeutics, on peptide synthesis, and they are much cheaper actually than the earlier time. Actually, the cost has reduced a lot, but I don't know how to reduce the cost further.
Okay. Thank you, sir.
Do we have any other questions here or from the YouTube? Okay, sir. Thank you very much, sir, once again for your insightful talk.
Okay, thank you. Okay, bye everybody.
Yes. So the next speaker will join very soon. Please stay with us.
I have joined. Okay.
Oh, yes, sir. Sir, can you please turn on your video?
I try to turn it on, but it's not letting me on. Turning me on actually. Yeah. Yeah. Now it is there. Yes.
Okay, sir. Yeah, good to see you, sir. Great.
Thanks.
So it is my pleasure to introduce the next speaker, Professor Sandeep Paul from IIT Guwahati. Very close to us, but because of the pandemic situations, he is unable to visit us. We would like to certainly have you next time in person, sir.
Sure.
So he is a professor in the Department of Chemistry, IIT Guwahati. He is a member of editorial advisory boards on many ACS journal, including JCIM, and also he is a bronze medal awardee of the Chemical Research Society of India. He has done postdoc from North Carolina University, USA, British Columbia from Canada. He has done PhD from IIT Kanpur. His research interest are molecular dynamic simulations of macromolecules, biological macromolecules and their complexes with each other as well as with small molecules. So we are very interested to hear about the applications of these MD simulations from him. And today he is going to talk about uric acid induced kidney stone formation and its disruption by vitamin C. Please, sir, over to you.
Okay. Thank you for your nice introduction. Okay. So let me share my video first. Okay. Let me first thank the organizer for inviting me for this nice conference.
Sir, your screen is not visible.
Yeah, I'm sharing it now. Is it visible now?
Yes, sir, we can see.
You can see it, right? Yes. Okay, great. Okay, good morning, everyone. The title of my talk is Uric Acid Induced Kidney Stones Formation and its Disruption by Vitamin C. So before going to the talk, let us see one video here that I have taken from YouTube.
Before we talk about how kidney stones are formed, take a moment to become familiar with the urinary tract. The urinary tract includes the kidneys, ureters, bladder, and urethra. Now let's enlarge the kidney to get a closer view. Here's a cross-section of the kidney. Urine flows from the outer cortex to the inner medulla. The renal pelvis is the funnel through which urine exits the kidney and enters the ureter. As urine passes through the kidneys, it can become very concentrated. When the urine becomes too concentrated, calcium, uric acid salts, and other chemicals dissolved in the urine can crystallize, forming a kidney stone or renal calculus. Usually, the calculus is the size of a small pebble, but ureters are very sensitive to being stretched, and when stones form and distended, the stretching can be very painful. Often, people may not know they have kidney stones until they feel the painful symptoms resulting from a stone being stuck anywhere along the urinary tract. Fortunately, small stones typically pass out of the kidneys and through the ureters on their own without causing any problems. However, stones can become more problematic when they block the flow of urine. Doctors call this one a staghorn kidney stone, and it is obstructing the entire kidney. Fortunately, these stones are the exception rather than the rule.
Okay, so as we have just seen that, okay, so uric acid actually is one of the main culprit for formation of the kidney stones. Okay, so what is kidney stone? A kidney stone is a collection of minerals and salt that hardens. Okay, kidney stones form in the kidneys or urinary tract, the path that urine takes to leave the body. They can cause pain and blood in the urine, but treatment can help. Okay, so there are several factors that promotes kidney stones like lifestyle, diet, climate, occupation, etc. And then genetic aspect is also there here. Okay, and some of the promoters, like if we take sodium intake, I mean we have if our sodium intake is more, or dietary calcium or high acid intake. Okay, so kidney stone can form, and there are inhibitors also like citrate and magnesium. So there are two types of kidney stones in broadly. One is non-infections, or rather three. One is non-infection stones, infection stones, and genetic stones. Among non-infection stones, we can have calcium oxalate kidney stones, and then calcium phosphate and uric acid. And among in infection kidney stones, we have stu white, and genetic stones like cystine is also there. So our main focus is here actually. Okay, so kidney uric acid induced kidney stone. Okay, and the drug treatments for this is like citrates, sodium bicarbonate, allopurinol. Okay, so this is our interest here. Now, what is uric acid? Okay, uric acid is a waste product that is in our blood. Our body produces uric acid when it breaks down chemicals called purines. Most uric acid dissolves in our blood and passes through the kidneys. It leaves our body in our urine. So this is the structure of the uric acid. You can see it. Now, how do uric acid stones form? Okay, so if we have high levels of uric acid in our body, okay, then crystals start to form. These uric acid crystals combine with other substances in our body and create a solid mass. The mass keeps growing. It may stay in the kidney or move down the urinary tract and settle in the ureter. If the stones are very small, they may pass out of our body in our urine without too much of pain that we have just seen in the video. But if they don't pass, they cause urine to back up in the kidney, ureter, bladder, or urethra. That's when we get pain and other symptoms. So who is at risk of getting uric acid stones? So if we don't drink enough water, we have a higher chance of developing uric acid stones and other kidney stones. Other risk factors include chemotherapy, conditions such as obesity and type 2 diabetes, diet high in salt and sugar, family history of kidney stones, too much or too little exercise, weight loss surgery, taking certain medications such as diuretics and immune suppressants. So how common are uric acid stones? Okay, one may wonder. So researchers estimate that 1 in 10 people in the United States will have a kidney stone at some point of time in their life. They are more common in men than women. For men, the lifetime risk about 19%. For women, it's about 9%. Most people don't get kidney stones before age 30, but many cases can happen even earlier in life, even among children. Now, we have been working on this problem for last few years. So we have published quite a number of papers like the one I have shown here. So in this paper, actually, we describe the self-aggregation of uric acid causes the formation of higher order clusters. So in this paper, we basically have looked at what is the mechanism by which the uric acid forms a larger cluster. And then here also we went into more detail about the uric acid-induced kidney stones. Now, how do uric acid? So in those two papers actually, or we have many papers on that. So we actually observe uric acid molecules self-aggregate in water. So the mechanism is like so through uric acid hydrogen bonding. You can see that a lot of hydrogen bonding sites are there in one uric acid molecules. And then there are a lot of hydrogen bond formation takes place, okay, among uric acid molecules. Okay, so hydrogen bond, uric acid hydrogen bonding is one factor. And the second factor is uric acid pi-pi interactions. Okay, so this is also there. So what is our goal then? Okay, so we have large uric acid crystals, and our goal is to disrupt or break down the large uric acid crystals into smaller crystals of uric acid. And then we need to pass it out through the urine. Okay, this is our goal. So our approach should be, we need to break the uric acid hydrogen bond, as well as we need to break or disrupt the uric acid pi-pi interactions. Once we achieve these two things, then we can get smaller uric acid crystals from large uric acid crystals. Once we get the larger, smaller uric acid crystals, then the dissolution of uric acid crystal is another part, okay, that we need to take care of. So what my today's talk is based on this paper. We published this paper very recently, and this is the guy who did this work, Krishna Gopal Chattaraj. Okay, so he has just joined as a postdoctoral fellow in the MIT USA recently. So he's a very hardworking guy. He worked on this problem. So in this paper, actually, what we considered uric acid, and then we used vitamin C. This is the structure of vitamin C, and then we have used one graphene surface here. Okay, this is hydrophobic acid graphene surface, non-charged. We have used here. So as I mentioned that, okay, so we have used vitamin C in our study. What are the benefits of vitamin C? One may ask. For individuals suffering from cancers to a common cold, or vitamin C has been regarded as a preventive. High oral doses of vitamin C on renal clearance and excretion of uric acid have been examined as vitamin C improved renal uric acid clearance. But excessive intake of vitamin C may also be associated with the formation of stones. That we need to take care of. But again, we have also studied this part in this paper actually, and in today's talk we have also included that. So what is our goal then? Okay, so our goal is like the detailed molecular mechanism of the interactions between vitamin C and uric acid to explore the inhibiting action of vitamin C on uric acid aggregation in solution phase. A monolayer graphene sheet is used to examine the impact of uric acid absorption on it, and also the effect of vitamin C on adsorbed uric acid. The incorporation of the graphene sheet is done to simulate the kidney stone decomposition in the human body. And what is our expectation? Though the comprehensive modeling of an actual kidney stone is challenging, the present study may provide a theoretical perspective on the aggregation, adsorption, and elimination of uric acid in the presence and absence of vitamin C. So we have done classical molecular dynamics simulations wholly, okay, and this is a simulation detail. So we have used general Lambert force field parameters for uric acid, vitamin C, and carbon atoms of graphene sheet. For water, we have used TIP3 model, because this TIP3 model is well compatible with the gap force field. And 5 nanosecond equilibration is done in isothermal isobaric ensemble, or NPT ensemble, at 300 Kelvin temperature and 1 atmospheric pressure. And then we have run 200 nanosecond production simulation in NPT ensemble. And for shorter range interactions, we have used one cutoff 10 angstrom cutoff we have used. And long-range interactions are treated with TME method or particle mesh UR method. And a non-charged graphene monolayer with dimension 6 nanometer times 6 nanometer in XY plane is used. So in total, we have considered 14 systems here. Okay, so you can see that they are named as V0, V4, V8, etc. Okay, since this not represents number of or not 4, etc. represents the number of vitamin C molecules here. And you can see from V0 to V0 24 or V0 40 rather. Okay, so we have fixed number of uric acid molecule constant in our system. We have used 8 number of uric acid molecules in our systems, and we have increased slowly. We have increased number of vitamin C molecules in our system. Okay, for V0 system there is no uric acid molecule. No, there is no vitamin C molecule. For V4 there are 4 vitamin C molecules and so on. And we have kept number of water molecule fixed in our study. Okay, from V0 to V40. Okay, so and then we have considered 2 more systems, V0 8 and V0 24. Here only vitamin C is used in water. Okay, so we'll explain this one, why you have used this system. And there is no uric acid molecules in these 2 systems. And then we have considered 3 more systems at the bottom if you look at. So we have U16G, U16V32G, and U25V40G. And these 3 systems, for these 3 systems, graphene CT is used. Okay, first we focus on these 2 systems, particularly V24 and V40, because we have used higher concentration of vitamin C molecules here. Now, probable mechanism. Okay, so we have taken different snapshots for different systems from V0 to V24. V0 I have not shown here, and V40 also I have not shown here. In V0, all the uric acid molecules are uric acid molecules which are represented in blue color here. You can see the blue color here. So the blue color represents uric acid molecules, and black color represents vitamin C molecules. Okay, so V0, all uric acid molecules are aggregated, and V40. Okay, so the uric acid molecules are disrupted. There is no cluster. There is no cluster of uric acid molecules there. So that's why we have just I just shown your snapshots of 4 system from V4 to V24. Now you can see that uric acid molecules they form a kind of they are aggregated here. Okay, you can see in all systems they are aggregated. And what is the role of vitamin C here? Okay, you can see that vitamin C they do not break the uric acid clusters. Okay, what they are doing, they are just surrounding the uric acid clusters. Okay, so they are basically encapsulating the uric acid clusters. Okay, so the role of vitamin C here is like vitamin C does not break the higher uric acid clusters into smaller uric acid cluster. Rather, they are encapsulating the uric acid clusters. This is the first thing we observe. Now, if you look at closely, okay, you can see that there are some pi-pi interactions can be visible here. We'll show it in later slide. Okay, we have calculated whether we have also examined whether really there is pi-pi interaction present among the uric acid molecules or not. We have already examined that, but we'll show it. But from the snapshot also you can see that there are some pi-pi interaction possible here between the uric acid molecules. Now, so in order to quantify the clusters, how many uric acid clusters or uric acid vitamin C clusters are formed in different systems, we have used cluster structure analysis here. And then for formation of the cluster structure or to calculate the cluster structure actually, or to quantify the cluster structure, we set different criteria like distance criteria we have used here. And here is the data. Okay, so if you see again the blue color, this bar diagram in this bar diagram from V0 to V40, the blue diagram or blue color represents the size of cluster of uric acid in uric acid cell assembly. Okay, and the red colors and yellow colors, they represent the size of clusters of uric acid in uric acid vitamin C conjugate cluster and vitamin C in uric acid vitamin C conjugate clusters. Okay, so we can see that so we have here all the systems again I have shown here. So for V0 system we have 8 uric acid molecules, and we have calculated the size of different cluster at different time intervals. Okay, so 8 nanosecond interval we have calculated from, we have started from 184 nanosecond up to 200 nanosecond. We can see that, okay, all 8 molecules, they are forming, they are aggregating and they are forming large clusters of 8 uric acid molecules. And they do not break with time, okay, the time we consider. Now, if we come to from V0 to V4, we can see, okay, so as we incorporate 4 cluster here, okay, although we have used 4 vitamin C molecules. So what they are forming now, if you see the low red color here, you can see that, okay, so we have 8 uric acid molecule in uric acid vitamin C cluster, red one. And this blue, then the yellow one, it represents the number of, or the cluster of vitamin C in uric acid vitamin C cluster. You can see that, okay, so they are like around 4, okay. So all 4 are actually surrounding the 8 uric acid cluster. Now, if we keep on increasing number of uric acid molecules, and if we concentrate on V40, now you can see that, okay, again in V40, all uric acid molecules are not, I mean, there is breaking of uric acid cluster here, okay. So you can see the size of the uric acid cluster is much smaller, around 2 or 3, okay, like that. And then if you look at the yellow color, you can see that, okay, very high, 40, around 40 it is coming, or around 36, okay, rather. Okay, so number of vitamin C in uric acid vitamin cluster. Okay, so we have uric acid or we have uric acid clusters, but the vitamin C molecules, they are basically encapsulating, or they are surrounding the uric acid clusters that we have seen in the cluster structure. We have simulated 2 different systems, another 2 different systems like V08 and V024, where there is no uric acid but only vitamin C molecules are there. And you can see that although we have used for V024, 24 uric vitamin C molecules, but we can see that, okay, up to 8 more is forming here. So vitamin C self aggregation is not that great extent, okay, in water, and the concentration we have used. Okay, so now we looked at, as I mentioned before, that, okay, so the clusters can be formed through hydrogen bonding interaction as well as through pi-pi stacking. So we need to now consider, okay, whether really there are hydrogen bonding interaction present between uric acid, and uric acid vitamin C, or vitamin C molecules. For that, we have used widely used geometric definition of hydrogen bond. So we have used one cutoff distance, okay, and we typically the cutoff distance about donor acceptor distance has to be less than or equal to 3.5 that we have used in our study. And then donor hydrogen acceptor angle, okay, we consider as greater than equals to 120 degree. Typically, it should be around 180 or so. That is the the ideal case, but we are relaxing a little bit, so we consider 1 up to 120 degree, or 120 degree or more. So when these 2 conditions they satisfy simultaneously, then we say that, okay, hydrogen bonding or hydrogen bonds are formed. Now we calculated uric acid, vitamin C, and vitamin C hydrogen bond for different systems. Now we just look at the V40. Okay, so from V0 to V24, there is not much change in uric acid hydrogen bonds. Okay, so up to this system like V24, uric acid clusters they don't break. Okay, that you have also seen in here. Okay, there is not much breaking of uric acid clusters. Okay, but above that, when you used very high concentration of vitamin C, 40 uric acid molecule, 40 vitamin C molecule, you can see that there is disruption of uric acid hydrogen bond, and the number of uric acid hydrogen bond drops from 7.59 to 4.1. Similarly, uric acid vitamin C hydrogen bond, there is a sharp jump from 23 to 35 as we move from system V24 to V40. And we consider V08 and V024, okay, we can see that, okay, there are hydrogen bonds, but not that much, okay, only 2 and only 12 here. Then we looked into whether really there is pi-pi interaction present or not. Okay, in order to do that, actually, we defined one angle, theta 2. Okay, so the theta 2 are this angle is defined theta 2 like, you have one aromatic plane like yellow color and one aromatic plane like your this brown color kind of thing. Okay, and then we consider 2 vector normals. One is passing through this yellow aromatic plane, and another is passing through this brown aromatic plane. And then we consider the angle formed between these two normals, vector normals, theta 2. Okay, so when theta 2 is close to 0 to 20 or 160 to 80, 180, then we consider that okay, pi-pi interactions are present. Okay. And for this, actually, now we see that okay, for uric acid vitamin C here, okay, if you look at here, so for uric acid vitamin C, okay, so you can see that okay, so around 0 to 20 and 160 to 80, okay, so pi-pi interaction is present, and there are some portions here also, kind of broad kind of distribution this position, but mostly here. So uric acid vitamin C pi-pi interaction is there. Now vitamin C, you can see very broad kind of thing. Okay, very loosely you can say that okay, vitamin C pi-pi interaction is also there. But most striking feature is here uric acid pi-pi stacking interaction, and you can see that okay, it's located around 0 to 10 degree and around 170 to 180 degree here. So uric acid, yeah, there is strong uric acid pi-pi interaction. Next, we calculate preferential interaction parameter and interaction energy. So okay, so for different system, we can calculated uric acid vitamin C interaction energy. So we have decomposed the total interaction energy into electrostatic component and van der Waals component. So blue color represents the electrostatic component and the red color represents the van der Waals component. And you can see that okay, so uric acid vitamin C, this interaction is mostly favorable by electrostatic contribution, but van der Waals contribution is not negligible. You can see okay, so it's around here around 40 and then here around 70, okay, something like that. Okay, so both electrostatic and van der Waals interactions are present in uric acid vitamin C interaction, but electrostatic interaction is dominating over van der Waals interaction. And then we calculated preferential interaction parameter. This is represented as tau here, and we have to calculate tau. We consider Kirchhoff integral. So this is the expression for Kirchhoff integral. And here Gij is the radial distribution function here. And then we calculated Kirchhoff, we calculated Gij, and once we get Gij, we substitute Gij here, and then we calculated tau. Now, what is this tau UW? Okay, this tau UW represents rho times rho u is the number density of uric acid in that system, and times GEU minus GUW. GEU is the Kirchhoff integral for uric acid interactions, and GUW is the Kirchhoff integral for uric acid water interactions. Okay, so this tau UW, it represents whether uric acid interaction is favorable over uric acid water interaction or not. Okay, so if the value of this tau UW is positive, means GUU is more than GUW, so we can say that okay, uric acid is interaction is more than uric acid water. Or uric acid, their interaction they are interacting preferentially more than with uric acid water interaction. And similarly for UV is like uric acid vitamin C interaction and so on. Now you can see tau UW is represented here, and we can see that as we move from V4 to V24, we have shown only for two systems, and we can see there is not much change here. And this for tau UV, and then you can see yeah, there is considerable change from V4 to V24 as we move from V4 to V24. Okay, so uric acid vitamin C interaction is slightly more favorable here. Okay, getting interaction is more here compared to V4. And for tau VU, we can see that okay, it becomes negative. Okay, for V24. Okay, so if we move from V4 to V24, it becomes negative. Means uric acid vitamin C uric acid interaction is more favorable than vitamin C interactions. Okay. Then we calculated dimer existence correlation function. It's basically it considered to be the dimer existence correlation is evaluated by the use of the following equation here, and it is considered to be the autocorrelation function of a simple function beta ij here for a pair of molecules say I and J that can have value 1 when they are maintaining a preset distance criterion, and it goes to 0 if the distance criterion is lost for the very first time. Basically, it calculates how long one dimer is stable. Okay, and we consider different dimers like uric acid, vitamin C, and vitamin C, vitamin C. And here is the plot, okay, for different systems. So if we consider uric acid dimer DSCF, we can see that as we move from V4 to V24, okay, so the exists like the DSCF value or the time or the existence time is more here. Yeah, okay, so the cluster is stable for longer time. Uric acid cluster is stable, or uric acid dimer is stable for longer time as we move from V4 to V24. And now you consider UA and vitamin C cluster. You can see that, okay, so no, it decomposes faster rate, or slower rate here. There is not much change, but it decomposes faster, slower rate for V24 system. And vitamin C, there is not much change. Okay, now we can, so from there we can see actually, okay, so this uric acid vitamin C, this dimer existence, correlation time, or the dimer, once it is formed, okay, it lives for a longer time. Okay, once we move from system V4 to V24. So yeah, so I can skip this, but it's not required. Then we can see, okay, effect of dilution. Okay, so you can see that, okay, so we calculated energy values here and the size of clusters, okay, etc. And what is striking here that, okay, so if you consider uric acid vitamin C interaction here, so as we increase the number of water molecules, you can see as we move from system V16 to V16B, we have kept number of uric acid molecules and number of vitamin C molecules fixed, and we have increased water molecules. Now, as we move from V16 to V16B, we can see that uric acid vitamin C molecules become less favorable compared to compared to V16 system. Okay. Now, next. So the large elongated cluster of uric acid is turned into small thin cluster in the presence of theobromine. Theobromine is another inhibitor that can inhibit or that can disrupt the uric acid clusters. So in this study, okay, they have found that, okay, so large elongated clusters are formed by uric acid molecules, and once theobromine is introduced into the system, these elongated clusters of uric acid is turned into small thin clusters. And we also looked at theoretically, okay, whether really this is what is the mechanism and what is why it is how it is acting, the theobromine molecules. And we have also found that, okay, large elongated cluster of uric acid is becoming more smaller and smaller uric acid cluster in presence of theobromine molecules, and we reported here in this paper. So in view of this, so now we are interested into, okay, so we have the cluster. Now we would like to see whether the application of vitamin C really breaks the elongated uric acid clusters or not. And for that, we have considered another system here. So we have used 16 uric acid molecules and 32 vitamin C molecules and 6,000 water molecules. And you can see the uric acid molecules, they form elongated kind of structures here. Okay. And again, you can see, okay, no, I mean, the vitamin C molecules, they do not break the elongated structure of the uric acid molecules. Okay, rather they are again encapsulating the uric acid clusters. Now there comes the most important thing. Okay, so the kidney stones actually, which are associated to be free or appended to renal papillae, are crystal precipitates in the renal calyces and pelvis. And they contain organic and crystalline constituents produced by the supersaturation of the urine. The most familiar type of kidney stone is formed on the renal papillary surface. Urinary crystals stick to create tiny hard mass of stones known as crystal growth. The growth of stone is achieved on the matrix coated surface with the aggregation of preformed crystals or the secondary nucleation of the crystal. So in view of this, actually we simulated uric acid vitamin C molecules in presence of graphene layer. Okay. So we consider three different systems. We use 16G, use 16 V32G, and 25 V40G. And you can see the number of uric acid molecules in first two systems 16, but in first system there is no vitamin C molecules, and the second system we have introduced 32 vitamin C molecules, and number of water molecules are fixed in both cases. Now, if you see here, okay, so you can see that, okay, so uric acid molecules, they are basically getting adsorbed at the surface of the graphene sheet. Now, for this system, 16V32G, we have now 16 uric acid molecules and 32 vitamin C molecules. Now you can see here that, okay, so uric acid molecules, they are basically getting adsorbed on the surface, and then again vitamin C molecules, they are surrounding the clusters of uric acid molecules here. Okay, and same is true for the other system also. Okay. Now, okay, so so far we have found that, okay, uric acid molecules, they form large clusters, and vitamin C molecules, they don't break the cluster, rather they encapsulate the uric acid clusters. Okay, so now next goal is, okay, we need to solubilize it. Okay, the uric acid vitamin conjugate clusters. Okay, so in order to do that, we calculated number of uric acid molecules. So UA is we have calculated the first cell coordination number. So it basically represents, okay, we have one reference uric acid molecule or a reference molecules, and how many molecules are surrounding that molecule. So UA here represents, we have one reference uric acid molecule, and then how many uric acid molecules are there in the first solvation cell of the reference uric acid molecule. And we can see that as we move from system B0 to B24, there is not much change in the coordination number value. But if we consider uric acid vitamin C, you can see that, okay, there is increase in vitamin C molecules around uric acid. And most striking feature is here. Okay, so for B0 system, we have 9.5 number of on average water molecules around a single uric acid molecules. But as we increase the vitamin C concentration, okay, you can see that, okay, water molecules are expelled out basically, and number of water molecules in the first solvation of uric acid decreases from 9.5 to around 5. Okay, and then we calculated uric acid water here. Okay, so you can see that, okay, again uric acid water hydrogen bond, this is hydrogen bond number decreases as we increase the vitamin C concentration. So next we calculated Fourier-Huygens interaction parameter. Why do we need to calculate Fourier-Huygens parameter? Okay, so one drawback or one limitation in molecular dynamics simulation is the solubility of a solid molecule in a solvent cannot be calculated directly in simulation. Okay, that is the main one of the limitation in the molecular dynamics simulation. That's why we took help from Fourier-Huygens interaction parameter. So Fourier-Huygens theory is a lattice-based model theory, and it was developed to understand the non-ideal behavior of polymer solutions. This theory assumes that the arrangements of molecules in the solution are completely random. The formation of solution depends mainly on the transfer of polymer chain from a perfectly ordered pure state to a much disordered state, and the process of mixing of the polymer chain with the solvent molecules. And the critical value of Fourier-Huygens interaction parameter we call chi FH for separation of a polymer solvent mixture is given by this expression. So polymer solubility criteria is for monomeric mixture, X is 1 and the value of chi is 2. And when X goes to infinity, X or chi FH becomes 0.5. Okay. Thus, when X or chi FH is less than 0.5, we can say that polymer is soluble. But here we have used the modified Fourier-Huygens theory. Why modified Fourier-Huygens theory? Because the modified Fourier-Huygens theory allows to consider intermolecular interactions between the molecules that are present within the mixture. And it also allows for the determination of chi FH as concentration changes. Those are the limitations in the original Fourier-Huygens theory, but modified Fourier-Huygens theory, it takes care of the molecular interaction as well as it takes care of the concentration change. So we calculated this Fourier-Huygens interaction parameter by this expression. Here V reference is the molar volume between uric acid and vitamin C, and delta E mixing is the energy of mixing between uric acid and vitamin C. Okay. Since the volume change due to mixing is insignificant in our case, so therefore delta E mixing can successfully be replaced by the enthalpy of mixing. So we replace delta E mixing by enthalpy of mixing delta E mix. And delta E mixing is calculated through this expression. Okay, so at the volume fraction UA and vitamin C in mixed systems are 5 UA and 5 vitamin C. Okay, and V represents the overall system volume. So we have used this expression for calculating delta E mix. Once we get delta E mix, we substitute it, we use this delta E mix in this expression, and we calculate as chi-FH or a Fourier-Higgins interaction parameter for different systems. And here is the Fourier-Higgins interaction parameter for different two systems we consider. One where uric acid and vitamin C ratio is 1:24 and 1:6. For 1:24 uric acid vitamin C ratio, we see that okay, delta E mixing is favorable, -13.35, and chi-FH is also negative. And when we increase the uric acid vitamin C ratio from 1:24 to 1:6, we see that okay, delta E mixing is much more negative and chi-FH is negative. So this chi-FH is since it's much smaller than 0.5, so we can say that okay, so the uric acid vitamin C, they are miscible with each other. So now this is the overall conclusion here. So basically we have found that the encapsulation of uric acid clusters by vitamin C molecules be false. The more vitamin C is present in a system, the more extensive interaction between the aggregated uric acid cluster and the vitamin C molecules. Vitamin C is very soluble in water. Uric acid clusters can be extracted from our body by applying vitamin C. Now there are certain limitations of this work because perfect modeling of kidney stones not possible. So we could not do it, and we did not consider the effect of pH here. Okay, because of some other reason. Okay, so these are the two limitations of this work. And finally, I acknowledge IIT Guwahati and then DSTSCRB and DBT for funding. And I also acknowledge Paramission Supercomputation Facility, IIT Guwahati. And thank you for your kind attention.
Thank you very much, sir, for a wonderful presentation and helping us understand the interactions between uric acid and vitamin C. I myself is very intrigued by this work and therefore have a question. So, did you, sir, consider including ions, potassium chloride, sodium chloride?
Not really in this case, but we have used another system where we have used K3C actually. Okay, citric acid, potassium citrate we have used there. Okay. But definitely we have not used any sodium chloride kind of thing here. Those ions are not present here.
Because they are expected to be the native environment.
It is there because it makes the system more complicated. Okay.
Yeah.
That's why we just not used it.
Okay, understood. So, any other questions from the audience?
I could see one question in the chat box. Okay. Okay, so the question is there any specific reason why the cutoff distance between donor and acceptor atom is taken 3.5 angstrom? Okay, so here I did not mention actually the cutoff distance is basically you cannot use randomly. Okay, so cutoff distance usually what we do, we calculate radial distribution function. Okay, it gives you, okay, so the probability of finding, suppose if you consider water, so we have one reference water molecule, and if you calculate water oxygen radial distribution function, then it says, okay, so you have one oxygen of water molecule, reference water, and then how many of water molecules are there in the first solvation cell. That gives you the first minimum of that GR. Okay, and we take that one. Okay, so that is the, basically we consider, okay, so number of molecules that are present within that distance. That gives you the first minimum of the GR of your radial distribution functions, and that distance we use for cutoff. Okay, and how will the change? Yeah, so as I mentioned, okay, if you increase that distance value, the number will change obviously, but we should not do that practice actually because we consider the molecules which are present in the first solvation cell. They only form hydrogen bonds with the reference molecule. Is it okay, Sanjana? I don't know. Yeah.
Yes, sir. Yeah, I believe so. So thank you very much, sir. If there are no other questions, we once again thank you for taking out time from your busy schedule.
Yeah, thanks everyone. Thanks. Yeah. Okay.
Okay, so we already have our next speaker with us, Professor Kunal Roy. Sir, can you hear me?
Yes, I can hear you.
Good morning, sir. We are very lucky to have you here with us. And Professor Kunal Roy is ex-head at the Drug Therapeutics and Chemoinformatics Laboratory, Department of Pharmaceutical Technology, Jadavpur University. He is among the top 2% scientists in the latest database, and he has been the editor-in-chief of International Journal of Quantitative Structure Property Relationships. He has been member of advisory board, editorial advisory board of European Journal of Medicinal Chemistry and many other journals. He has received Marie Curie Fellowship from and visited University of Manchester, UK. And his research interests are QSAR, molecular modeling, chemoinformatics, ecotoxicological modeling, and applications of AI in biomedical sciences, big data mining, and healthcare. So, and he is today going to talk about chemical read across predictions for toxicity and ecotoxicity data sets, which is very in line with the workshop theme. So with this brief introduction, I hand it over to Professor Kunal Roy. Sir, please.
Good morning. Can you view my screen?
Yes, sir. Please go ahead.
Okay. So at the outset, I would like to thank the organizer for inviting me here for delivering this talk at this conference. And the title of my talk is Chemical Read Across Predictions for Toxicity and Ecotoxicity Datasets. So in fact, we are surrounded by chemicals. We have so many chemicals in industrial use. We have so many chemicals in domestic use in everyday life. And we can categorize these chemicals as organic chemicals, pharmaceuticals, cosmetics, biocides, nanomaterials, ionic liquids, and so on. Now, the effect of all these chemicals on different ecological species is a matter of great concern because these chemicals will have toxic effects on biological organisms and through the food chain that also come up to human through a process that is called bioaccumulation and also biomagnification. The effect of hazardous chemicals and pollutants on the ecosystem is a matter of great concern. And as I mentioned, we have a large number of chemicals and large varieties of chemicals around us, and new chemicals are entering the market, into the market at a very high rate. So our human and material resources are insufficient to obtain experimentally even the basic information on environmental fate and effects of all these chemicals. So we need to use some modeling methods to know toxicity potential of these chemicals to bridge the data gaps in absence of experimental data available. Now, this kind of in silico modeling methods are time and cost effective, avoiding animal experimentation, and they support 3R principles: replacement, reduction, and refinement in animal experimentation, thus complying with the ethical requirements. And these kind of approaches are also supported by different organizations like European Center for Validation of Alternative Methods, International Organization of Medical Science, US EPA, OECD, Organization for Economic Cooperation and Development, and also it complies with the REACH regulation in the European Union. Now, among different in silico techniques in regulatory toxicology, we usually use two main methods. One is chemical read across, and another is quantitative structure-activity relationship studies. Both these techniques are based on chemical similarity measures. Today, I will focus on read-across. Read-across is a prediction method for unknown chemicals from chemical analogs with known property value. I would say this property may be toxicity, may be activity, or maybe some physiochemical property also. So this is basically a similarity-based approach, which is accepted by EU REACH and US EPA for data gap filling. This is defined as a method where we will use chemically similar compounds to predict the properties of the query compounds. And this may be of different types, like we may apply one-to-one strategy, one-to-many strategy, many-to-one, and many-to-many. In case we have a limited number of source compounds, then we apply analog-based approach. And if we have a considerable number of source compounds, then we will consider that as category-based approach. Now here, we can see the example of analog-based approach where this is the query compound and this is the source compound. So based on their similarity in the descriptor space and depending on the required feature important for the particular endpoint, we can predict the property of the query compound from this source compound. In case we have a number of compounds available, in that case, we can apply category-based approach, and both these examples are based on interpolation. But sometimes we can also a method called extrapolation, where the query compound is outside the descriptor space or descriptor range of the source compounds. Now, what is similarity? Now we express molecular structure information using different kind of representation. It may be 2D or 3D, but basically these are picture or graphics. So we have to convert the information present within the graphical representation into some numbers so that we can do numerical manipulation. Now the information, when stored in the form of numerical values, these are called descriptors. Now, once we get descriptors, we can do different kind of application of statistical and geometrical applications to know important features required for the particular endpoint. And we can do different kind of learning exercise to develop a model, and finally, we can predict the endpoint of interest. Now, molecular structure information can be stored through number at different dimensions. It may be 0D, maybe simple constitutional descriptors. It may be 1D based on some substructures. It may be 2D based on graph theoretical approaches. It could be 3D based on representation of molecular structure in three-dimensional form. We can also apply our conformational aspect in 4D. Now, whatever is the method of calculation of descriptors, but basically we derive a matrix that is called descriptor matrix like this. Say, for example, if we have chemical structures, simple chemical structures like these, we can calculate some molecular descriptors. That means some information about molecular structures, which may be calculated at different levels of theory, maybe at very low level, say information like molecular weight, number of carbon, nitrogen, etc. That also convey information about molecular structure. But we can calculate descriptors at higher levels using different software. Now we apply data pre-processing for normalization, standardization, feature selection, and identification of outliers. And finally, we can trim down the size of the descriptor matrix, and then we can apply appropriate feature selection strategies to understand which features are important for our particular response that we are going to model. And then we can apply a different statistical tool for model development, which may be either regression-based or classification-based. In case our endpoint is continuous in nature, we can apply a regression-based strategy to derive a model which will be able to predict the endpoint in a continuous form that giving continuous values. And in case we do not have our endpoint values in a continuous scale, but we only have graded values like toxic and non-toxic, or active or inactive, we can go for development of a classification-based model. And finally, whatever model we generate, we should check the statistical quality and applicability of that model through various validation metrics, quality and validation metrics. I have mentioned about similarity. Now, in the simplest sense for read across, if we consider that there is only single molecular feature determinant that is responsible for our response, then what happens? So in that case, say we have two source compounds and only one query compound. So this is our query compound, and we have two source compounds, and in the descriptor space, our query compound is within the two source compounds. In that case, we can define slope of this line based on the source compound 1 and query compound, and also from source compound 2 and query compound, and then we can equate these slopes. And from the reorganization of this equation, we will be able to calculate the Y0 value, that is the response for the query compound. But in case our query compound reads beyond the descriptor range of our source compound, that is also possible. In that case, we calculate the slope from the two query compounds, and then we calculate our response from the compound which is closer to the query compound, and using the slope that we have calculated from the two query compounds. So this is the simplest example where we have only one molecular determinant for our response. But in most of the cases that doesn't happen. We will have several features. So we need to reduce the dimension of those features. And one of the methods is principal component analysis, where we can reduce the dimensionality of our useful descriptor matrix to a lower dimension without significantly losing information. And then if we plot principal component 1 versus principal component 2, then we'll see clusters of compounds showing similarity among them. So that will be helpful for identifying similar query compounds to similar source compounds. Another method may be self-organizing map, which is a unsupervised method of that is mapping. It's unlike the neural network. We are not using here feed forward because we are not using our response values, but from the pattern of the X descriptors only, we'll be able to find out some pattern in the set of compounds, and we can identify from the surface map the close compounds, close query compounds to the corresponding source compounds. Now, if we go back to QSR to some extent, because QSR is also a similarity-based approach where it develops model by correlating biological activity or any other property of chemicals using descriptors by application of statistical tools. So the basic notion of any QSR is that the biological activity or the response property is a function of different molecular features, which are designated here as X descriptors with their corresponding regression coefficient values. So this is an example of regression equation in the simplest form as multiple linear regression. And when we plot our observed values versus predicted one, in case of a good model, there should be a low degree of scatter, and our point should be close to the 45-degree angle line. Now, we have to measure the quality of the developed model by using different kind of metrics, and the simplest metric is determination coefficient, where we compare the sum of squared residuals, that is difference between the observed and model derived values in squared form, and if we take sum of those for all training compounds, and this measure is compared to some reference. Now, in case of no model, we have the reference as mean response value of the training set compounds, and we can find out the individual difference between the observed values and the mean of training compounds, and we take square of those, and we can take sum of those for all the training compounds. Then obviously the numerator will be smaller than the denominator because it is a comparison with no model. In that case, R square value will be 1, and square root of R square is correlation coefficient. Now, the correlation coefficient does not give any information about the number of descriptors used in a model, but for a meaningful model, it should have a good degree of freedom. So number of data points should be higher and number of descriptors should be lower. So to penalize a model with higher number of descriptors, we can define explain variance, where we also use the number of descriptors used in this expression. We can define variance ratio as a measure of stability of regression coefficients and a measure of significance of the correlation coefficient. And we can calculate standard error of estimate, which should be a low value for a good model at a particular degree of freedom. Number of compounds minus number of descriptors minus 1. But the previous measures do not give any idea about the applicability of the model on new set of data, because those are based on fitting exercise. Now, to apply a model on a new set of data, we have to check validation of the model. The validation can be done using several methods. I'll discuss about 2 methods here. In case of internal validation, it can be done by cross-validation using methods like leave one out or leave many out, where one or some percentage of the compounds from the training sets are removed and model is regenerated from rest of the compounds, and using that model, the omitted compounds are predicted. And this is done in iteration. For all the compounds, we will get the predicted values, and then we apply this expression, which looks like similar to determination coefficient, but this is called cross-validated R square or Q square, and this value should be higher than 0.6. Now, in case of external validation, we apply a different set of data where we develop our model from the training set, but apply that model on a different set, that is test set. Test set is not used during model development, but we know the response values for the test set compounds. So one way of checking the applicability of the model on new set of data is by applying the model on the new set of data, and then we can compare the model-derived prediction with the observed values. And this expression is also similar to R square. The only difference is that here we are measuring the difference between the observed and predicted values for the test set compounds, and we are comparing those to the difference between the observed values of test set compounds and mean value of the training set compounds. So this measure is called Q square external F1, and there is another variety where we can compare this with the mean value of the test set compounds. In that case, it is called Q square external F2. Now, this measures are statistical approaches. So this external validation measure we also require in case of didacross. But in case of didacross, there is no model. There is no statistical model development, unlike QSR. But once we develop our hypothesis and get some predictions, then we can use those predicted values for calculating these validation metrics to know the quality of predictions and applicability of the model for new set of data. Now, in case of QSR, it is a statistical approach, so we have to follow some basic steps. For example, in case of biological QSR, we have to use logarithmic conversion of the response values. Then we calculate descriptors from chemical structures, and we do a data pre-treatment to remove intercorrelated descriptors, etc. Then we divide the data set into 2 parts, training and test. As I've already mentioned, training is used for model development through a process of feature selection and application of chemometric tools. Then once the model is developed, then we can apply the model on the test set by checking the applicability domain. This is very important because all models are not suitable for all test or query compounds. The test or query compound should be similar structurally to training set compounds. We cannot use a completely different chemical city is for prediction using a QSAR model that has been derived from other chemical category. Some sort of similarity must be there, otherwise QSAR model is not applicable because it is based on similarity principle. The applicability of QSAR models is judged by different quality and validation metrics that I have already mentioned. And once we are satisfied with different quality and validation metric values, then we can go for interpretation of the model for mechanistic explanation for its diagnostic use, and also we can go for design of new compounds. Now, as QSAR is a statistical model development process, we have to think about the statistical factors, like we should have a large number of compounds and lower number of descriptors so that our model is statistically sound. If we are using a very small data set and if we are using a large number of descriptors, that is statistically not meaningful. In fact, if we use the same number of descriptors as is the number of data points, we will get R square equal to 1. But that doesn't mean that we have got a good model, because when you calculate explained variance, that will show that the model is penalized due to large number of descriptors. And also you have to calculate different validation measures, etc. So in case of QSAR, we have to think about those statistical requirements. But in many cases, we may not have sufficient number of data points. We may not be able to develop a statistically sound model. So what we do in those cases, if our response values or endpoint is very precious, we need to use some model for data gap filling, for bridging the data gaps. But if we cannot develop statistically sound model, then what we do? In those cases, chemical read across is very useful. In some cases, it may so happen that we have a very limited number of compounds, but we need to have some kind of prediction for our query compounds. But in absence of model, we can apply read across. QSAR is not applicable for small data sets. Read across is not a statistical fitting process. Calculation is comparably easier than QSAR. Alternative tool, it can be used for alternative tool for hazard assessment, aiming at filling of data gaps. For example, here I will give one example of nanotoxicity estimation. In most of the cases, these data sets are very small. Usually, we do not get more than 17 to 20 number of data points. So it is difficult to develop meaningful model, because we have to also go for training and test division. In that case, we are losing some information. Training set is small, test set is also small. We are not confident about the statistical quality and applicability of the model. So there is a problem with QSAR. So we can use here chemical read across. So the case study that I'm discussing here is for development of an easier and efficient method of quantitative read across, which can be used for various small data set. We also compare our results with previous methods, and also we have developed an application for read across predictions. Here, 3 different data sets are taken from previously reported work. All 3 are nanotoxicity model development. Well, the first one is against human keratinocyte cell line. Second one is against E. coli, and third one is against E. coli under dark condition. We have used the same features that have been reported previously. Like for the first one, we have used the molecular electronegativity measure of the cluster and enthalpy of formation of metal oxide, etc. And for the second one, enthalpy of formation of gaseous cation and charge of metal cation. And for the third one, enthalpy of formation of gaseous cation and absolute electronegativity of metal oxide. Now, what we do in all the cases, we have a number of data points less than 20. We divide into 2 parts, training and test. Although training and test, the names are not appropriate here, we will use the names source and target compounds. So we divide equally. So we take 50-50 ratio for source and target compounds. Then we use 3 different similarity calculation methods: Gaussian kernel, Laplacian kernel, and Euclidean distance-based similarity. We sort the training compound for each query compound in descending order and select up to 10 most similar training compound for each query compound. As I've mentioned, we have here less than 10 compounds in the in the list of source compounds. So we use as many compounds are available here, and then we give weightage to the similarity, and finally we calculate the predicted value of the query compound based on these weighted average predictions. So the first one, the Gaussian kernel function, is obtained from this expression where x and y are two input vectors, and this is the Euclidean distance or L2 norm. And sigma is a hyperparameter here, which is to be optimized against different values, like say 0.25, 0.51, 1.251, 1.5, etc. The second one is Laplacian kernel function, which is obtained from this expression where we use the Manhattan distance between two vectors, or L1 norm, and gamma is a hyperparameter to be optimized. In case of Euclidean distance-based similarity, we do prior scaling of descriptor values using the mean value of the training set compounds and their standard deviation. And using that scaled values, we calculate the Euclidean distance of individual test compound to training compounds. And finally, we also convert the calculated ED into 0 to 1 scale. And finally, we calculate similarity from 1 minus scaled Euclidean distance. So now we have three different similarity measures: Gaussian kernel, Laplacian kernel, and Euclidean distance similarity. And once we get our predictions from weighted average prediction calculation, using those predicted value, we use our previous validation matrix, Q square external F1, F2, those we have already discussed using this expression, and root mean square error of predictions. Remember that the previous part, the reductor part, is not statistical, but once we get the predicted values, then we apply statistics to get different validation measures. In case if we are doing classification, obviously we will not use regression-based measure. In those cases, we will use our usual metric for classification, like sensitivity, specificity, precision, accuracy, F measure, G means, Cohen's kappa, etc., from the information of true positive, true negative, false positive, false negative, etc. Now here we have developed a Java-based application which is available from this site. It needs to input file training and test in the Excel format, and also user has to provide the sigma value and gamma value. The usual starting point is 1 in each case, and also number of similar training compound that the user wants to use, that also to be given, and distance threshold and similarity threshold. And the program generates two output files. One is the predicted response, and another is sorted experimental response with respect to distance and similarity. These are the different snapshot of during work of the software. This is one of the output files that shows the sorted training compounds for each query compound, and this is the predicted endpoints based on the three methods. One is Euclidean, second one is Gaussian kernel, third one is Laplacian kernel. And from these values, we are able to calculate different validation metrics if the user can provide the response values for the query compounds also. And these are the different metrics for external F1, F2, and RMSE-P. Now, from the first run of our experiment, in case of dataset 1 and dataset 3, we got acceptable results for Q-score F1 and F2, but dataset 2 shows somewhat lower values. So now we have to optimize the different hyperparameters and other settings. Now we have gone for hyperparameter optimization. In case of dataset 1, it showed the best results in case sigma was 0.75. In case of dataset 2, the best results are obtained when sigma is 0.5, and in case of dataset 3, again, sigma is 0.75. In case of Laplacian kernel, again, we need a different setting, like gamma equal to 1 for dataset 1, 2 for dataset 2, and 1.5 for dataset 3. Also, we have studied the effect of number of close training compound for toxicity prediction. And we have seen, actually, we do not need all 10 or close to 10 compounds. Sometimes the lower number of compounds, like 5, and in some cases 2 also give the best results. So this is because all training compounds are not required for prediction of a particular query compounds, because different training compounds are at different level of similarity. So when you are using all 10 compounds, that means there may be so many compounds which are less similar or more dissimilar to the query compounds. In that case, they are contributing negatively to the prediction. So this is also to be optimized. This is the graphical representation of optimization of different parameters at different settings. Okay, now we optimize the distance and similarity threshold value. And we have seen that for different data set, we have got different required thresholds, distance and similarity threshold. For data set 1, it is 0.4, distance threshold is 0.4, and similarity threshold is 0.05. In case of data set 2, it is distance threshold is 0.5 and similarity threshold is 0. And in case of data set 3, distance threshold is 0.4 and similarity threshold 0 to obtain these measures, best measures, which are also shown diagrammatically through radar plot here. Also, we have gone for classification-based measurement. If we take the same data set for classification-based measurement, taking mean value of the training compounds as the threshold, we see that in case of dataset 2 and dataset 3, at the optimized setting, we are getting 100% sensitivity, specificity, accuracy, precision, etc. for the query compounds. In case of dataset 1, we get somewhat lower value for sensitivity and accuracy. Now we compare this read across prediction with the previous methods, previously reported methods. Now our methods results are as optimized setting. So for dataset 1, we get Q-score F2 0.96 versus previously reported quantitative read across 0.74 and nano QSR 0.83. In case of dataset 2, Q-score F2 in our case 0.91 versus previous quantitative read across based on principal component analysis 0.80 and nano QSR 0.83. In case of dataset 3, our method gives 0.95 versus previous method of read across 0.91, and nano QSR is negative, showing negative value for Q-score F2. So based on this part of work, it is evident that this read across algorithm can predict the nanotoxicity of small data set efficiently, and all these reported cases, these values are actually better than corresponding reported QSR models. This works based on similarity-based algorithm, and we have used 3 similarity functions: Euclidean distance, Gaussian kernel, and Laplacian kernel. We have optimized different hyperparameters and number of close training compounds. Also, we have used different thresholds for distance and similarity measures, and this application is available from this site free of charge. And it can be used not only for metal oxide toxicity, but any kind of property or toxicity. We have also worked additionally for other sets which are yet unpublished. For example, the androgen receptor binding affinity, we had 147 compounds. This is not small data set, it's a bigger one, but we wanted to see how it works for a larger data set. So initially we had divided the data set into training and test. Then training is also divided into sub-training and validation set to optimize the different hyperparameters. And using these settings, we have applied for the test set and gone for chemical read across prediction. And we have seen that the Q-score F1 and F2 are showing the acceptable result. Actually, for this particular data set, the results are better than the corresponding QSR results. In another case, we have applied these for soil ecotoxicity estimation against Falsomia candida. In the same manner, we have divided the data set of 45 components into training and test. Training is again divided into sub-training and validation set. We have optimized different settings, and using one of the optimized settings, we have applied it for the test set. And finally, we have got the results like this. As you can see, that the Q-score F1 and F2 value are optimum in case of Euclidean and Gaussian kernel-based division, similarity measure. Also, we have used this for anti-COVID data set, that is antiviral activity against SARS-CoV-2. Here also we have divided the data set into training and test, and training is again divided into sub-training and validation set here. And we have gone for hyperparameter optimization. And finally, the results are like this for using different features. We have got results, and you can see the Q-score F1, Q-score F2 values. These are going up to above 0.9, and in all cases is better than corresponding QSR models. So all these examples show that chemical read-across is a very efficient method of predictions, and sometimes the results are better than QSR, and this is very good for smaller data set, because in case of smaller data set, we are unable to develop statistically sound models. So this is the snapshot from the website for the program. The nanotoxicity work that I have discussed here as case study, that was published in Environmental Science Nano recently. And this work was supported by ACRB. These are some of the text and reference book for understanding QSR and read-across for the beginner. And these are some of the reference book for different application of QSR in pharmaceutical, chemical, food, agriculture, and environmental sciences. These are more recent application of QSR in different diseases like Alzheimer, multi-target drug design, drug repurposing, and also for ecotoxicological QSR. And these two are more recent: in silico modeling of drugs against coronaviruses, and application of chemoinformatics and chemometrics in aquatic toxicology. So with this, I end my presentation. So in case of any question, I'll be happy to answer.
Thank you very much, sir, for a wonderful talk. And yes, we believe that these read-across methods are also will have applications in the drug discovery projects, where the number of compounds is limited, and as you said, that the statistically sound model may be difficult to build. Therefore, these should be looked into seriously by small drug discovery groups.
Yes.
So if you could like to elaborate on that, it will be great.
Yes, in case of drug discovery, we usually use QSR, but in regulatory toxicology, the read-across is more used than QSR, and it's more accepted to industries and regulatory bodies. Because in many cases, what happens that the number of source compounds are very limited, so it is difficult to produce QSR models which is statistically sound. So, and the method is also much easier in case of read-across. So in case of regulatory toxicology, chemical read-across is used more than QSR. But in case of drug discovery projects, read-across is not so far that much common, but it is actually growing more interest, especially considering biological read-across, considering the network of different diseases, network pharmacology, and different pathways contributing to particular disease. In case of multi-target drug development, the biological read-across is becoming very important.
Okay, thank you, sir. So any questions that we have?
I think there are some comments. How we can test our model when reference experimental data is not available. Okay, if reference experimental data is not available, if your data is not there, then it is difficult to judge the quality of prediction. Yes. So in case if it is a regression-based model, QSR model, then we have a tool that is prediction reliability indicator that considers some features like applicability domain, the closeness of the predicted values from the training set mean, and based on the performance of the model for cross-validated predictions in terms of leave one out prediction error, we have some tool. But in case of read-across, so far in our version 3, which can also give the classification-based prediction, it also can generate read-across ROC curve, and you have AUC. And from that AUC, you have some measure how good or bad the model is for prediction purpose. But specifically for quantitative predictions, we are in the process of development of some measure to know the quality of prediction for the data point which doesn't have any experimental values. So we are in the process of developing that tool to judge the quality of prediction from reducers for a data point for which we do not have any observed value.
That sounds interesting. We would be looking forward to that.
Yeah.
And if you give us some early access to that, that will be also great.
Yeah, sure. Once we develop, but it's in the development stage. Actually, we are working on that. Okay, there is another, I think, how to develop a QSR model on data taken from different biological assay like enzymatic assay. Okay, so one of the requirement of QSR is that the biological activity values that you are using should have been tested using the same experimental protocol. Because if you are using different experimental protocols, obviously you will have different ranges of values for even for same compound. So in that case, QSR, quantitative QSR, becomes useless. So how you can incorporate? One way of incorporating is to do classification QSR. Because in case of classification, you are doing only the active, inactive, or toxic, non-toxic. So there should be a threshold for each biological assay. You have to define a threshold. You have to get active, inactive, or toxic, non-toxic, or whatever. Then you can go for classification QSR by clubbing the available data from different assays. So that is one of the methods. Okay. What point one should consider to minimize error in such QSR model? So this is what I mentioned, that you can go for classification-based QSR in such cases, where the biological assay has been performed using different methods. Maybe you have obtained the data from different laboratories also.
Yes, sir. Thank you so much. Do we have any questions on YouTube? Okay. Thank you so much, sir, for this wonderful talk and nice explanation about the read across method, which looks very interesting and promising even in the future.
Yes.
And certainly we will go through some of the books that you have also shared with the audience.
Okay.
Thank you very much, sir.
Okay. Thank you very much.
So we are just waiting for the next speaker to join. And we will be back online as soon as we have him with us. Hello, are we online?
Yes, sir.
Okay, welcome once again. So we have with us Dr. Devendra Dhakad from Naipar, Kolkata. He is one of our old friends. He is currently working at Naipar, Kolkata as assistant professor. He has done his M.Pharm from BCP Mumbai, PhD from Naipar, Mohali. He was postdoctoral fellow at the National Cancer Institute in USA. And also he did another postdoc from the School of Chemistry at University of Hyderabad. And his expertise are chemoinformatic tools in machine learning, drug discovery, applications of ligand and structure-based multidimensional QSAR for variety of drug discovery applications, protein modeling, as well as theoretical chemistry in the area of quantum chemical methods and QMMM interactions. Today he is going to talk to us about basics of molecular interactions in medicinal chemistry and drug design. Thus, I feel beginners in the area should be very attentive and listen to him carefully. With this brief introduction, I welcome you, Dhakadji. Over to you.
Yeah, thank you, Dr. Wahab, and giving me opportunity on this program you are conducting for 3 days. Yeah, Dr. Wahab, whether these slides are visible?
Not yet. Yeah, now they are visible.
Okay, thank you. So, good afternoon, everyone. And today I have the talk on the basics of molecular interactions in the medicinal chemistry and the drug design. So, bear me, I am not presenting any research work, but this will be very useful for the master students and the beginners in the PhD programs. So we know there are various kind of the interactions are reported in the drug and receptor or any other macromolecular targets. It may be like protein, enzyme, DNA, RNA. So these are the few common interactions people have identified, and we are commonly using them. And these different kind of interactions are available in different softwares. As medicinal chemists are also trying to optimize some of the popular interaction like hydrogen bonding interaction or ionic interactions. So we will see about these different kind of the interaction in today talk. So binding of any molecule with any target, it depends upon the what kind of the functional groups we have over on inhibitors or drug molecule, whether we have any complementary nature of the functional group present in drug and the receptor. Based on that, we have the different kind of the interaction. In addition, there are several steric factors, safe factors, which also govern their interaction. So mainly we have the intermolecular interaction. Whenever we have the intermolecular interaction, these are responsible for the rigidity in a molecule. Suppose if we have the intermolecular hydrogen bond, this is responsible for the rigidity in the molecule. So most of the whatever the interaction we see, these are the reversible in nature. Rarely we see these are the non-reversible. But nowadays there are several examples have come in the literature, or people are doing a lot of work where we see there is a formation of the irreversible bond between macromolecular target and the molecule of interest. So here we see this is a protein, and then green color, this is the active site where the you see one molecule is there which is sitting in the active site. So macromolecules has got a different pockets, and out of those several pockets, 1 or 2 pockets are responsible for their physiological functions. So out of several important interaction, first one we are going to discuss about covalent interactions. So this type of the interaction are usually because of sharing of the electrons between the interacting atoms. So because this is a covalent bond, one of the strongest bond, so energy associated for this kind of the interaction vary in the long range from -40 the around 110 kilocalorie per mole. So such kind of interactions are rare in the receptor. If any drug is binding to receptor, such kind of interaction are very rare, but few examples are there. But these type of interaction quite common in the drug and enzyme and the drug and DNA interactions. So to have a covalent bond between target and any inhibitor molecules, we should have the two type of the reactive group in the target as well as the inhibitor. So these are electrophile or nucleophile. So nucleophile already present in the different macromolecules like DNA, RNA, as well as in the proteins. In proteins, we have the some of the important residue like serine, cysteine, lysine, glutamic acid. And from we see about DNA, RNA side, we have the adenine and guanine. Their ring nitrogen are highly nucleophilic in nature. And if we talk about electrophilic centers, there are several electrophiles which may interact with the nucleophile, may lead to the formation of a covalent bond. So we know epoxide ring is quite electrophilic in nature. And recently there are several drugs which have come up which has got a Michael acceptor group, means alpha beta unsaturated group is there. So if we have such kind of the functional group present in either in the protein or our ligand, then there is chance that we can have the covalent bond formation. So here is one example of drug. It has got this electrophilic center, and one of the nucleophilic serine is interacting, cysteine is interacting over here, leading to the formation of the covalent adduct. So there is another example from the adrenergic class of drug molecule which has got a electrophilic, usually it is not present in the active form, but in the physical condition, there is a replacement of this chlorine by SN2 reaction lead to the release of the chlorine, formation of this highly reactive ring. This is 3-membered ring has got a positive charge over the nitrogen. So now this is highly reactive, so one of the cysteine is interacting or attacking over this ion, leading to opening of this and lead to the formation of the covalent adduct. So only few drugs are involving in the formation of a covalent adduct with the receptor. So here this is a representation of the GPCR, which are 7 transmembrane receptor, out of which TM3, helix 3 has got a several residue which participate in the binding of with different drugs. And in the case of the DNA binders, which also has got a similar kind of the molecular features which lead to formation of again that active 3-membered ring, and which any of the residue from the adenine and guanine may interact with this and lead to the formation of the DNA adduct, or we can say it is a DNA alkylation. So if we see the history of these kind of the molecules, around 130 or before, Bayer company from Germany, they have synthesized the aspirin. That was the first molecule, we can say clinical molecule, which are acting by this method, by the formation of a covalent bond with the COX enzyme. Then during the World War, people have come up with the one molecule penicillin. Then next class of the penicillin, cephalosporin C, and other antibiotics. Then potassium proton ATPase inhibitors. And nowadays, several drugs are there, especially from the kinase family, which are working based on the formation of a covalent bond. So if we see the literature, what kind of people have done research? If we see before 1990, if we see there are less number of the research was there, so we see less number of the publication. But if we see in the last 20 years, people are working a lot of in this area, so we see huge number of the publication are here. So if we see last 10-year data, so every year we are getting one molecule which are making a covalent bond with their respective targets. So if we see from 2011 to 2019, almost every year there is approval of one drug which are working by formation of a covalent bond. So similarly, there are some poisonous gases are there which are also working by making covalent bond with the enzyme acetylene choline esterase. So by chance, one German scientist was working for the identification or development of a pesticides. By chance, he found this molecule. Like 3-4 years back in the Syria, people have used this gas sarin for in the different war. So these are also examples for the covalent inhibitors. Next type of the interaction is the electrostatic interaction or ionic interaction. If we have the two opposite charge, permanent pole charge, bond charge, then we have the possibility of formation of such kind of the interaction. Compared to the covalent bond, these kind of the interaction are weaker in the strength, but even though these are quite common in the drug receptor interaction or drug macromolecular interactions. So to make a such kind of interaction, we should have some positive center or negative center in the macromolecules. So they are acidic amino acids which can form the anionic centers, and if drug molecule has got a positive center, so they lead to the formation of ionic interaction. And those macromolecules can also have the basic amino acids which in the physiological condition are present in the protonated form. And if drug molecule has got any anionic center, then we can have again possibility of formation of a cationic anionic interaction or electrostatic interaction. So here you can see there is one example is shown over here. This is the arginine from the one of the amino acid which is present in the protonated form, and this is the one of the antidepressant drug which has got a carboxylic acid group. So there is a formation of a ionic interaction. There is another example of norepinephrine. So this drug is binding to the GPCR class of the receptors. So this interact with the aspartate, which is the acidic with this amine group. In addition, I also make other interactions, but just I am giving examples for the ionic interaction in this slide. There are several other examples, like we know the acetylcholinesterase enzyme is there. So it has got an anionic center, which is recognized by this quaternary ammonium ion present in the acetylcholine. If we do not have, people have seen several isosteres which do not have any positive center, those are molecules are not active, which are not identifying the acetylcholinesterase enzyme. Similarly, there are other examples which are mainly toxins based on the guanidine ion. So these are binding to the upper surface of a sodium channels. Similarly, there are several local anesthetic agents which are the mainly weak bases. So they are crossing the neurons and going inside the neurons, and in the neurons they are protonated and binding from the inner wall of a receptor, lead to the closing of their activation loop or gate. So as you can see, in these all cases, only ionic species is interacting mainly through in the active site. Salt bridge interaction. So this is a special kind of the interaction which involve the two kind of the interaction together, electrostatic interaction as well as the hydrogen bonding interaction. So this kind of interaction is possible if we have the two charged species and one of the species have the hydrogens. So here you can see like glutamic acid and lysine are interacting by salt bridge interaction. It may be possible that if such residue are quite deeply buried in the active site, these may not be ionized. But when these do different kind of the residue or any charge species are coming together, so acidic group may deprotonated, and that deprotonated, so that proton will migrate to the amine group and lead to the such kind of the interactions. So recently there was one analysis on such kind of the interaction. They found out that there are possibility of two kind of the interaction in this category. In one case, acidic residue like aspartate glutamate working as a acceptor, and while the protonated amines from the ligand are working as a salt bridge donor. So to make such kind of the interaction, we should have angle around 180. So another possibility that if basic residue like lysine, arginine, histidine, which are present in the protonated form in the physiological condition, and if we have any ligand which has got any anion like carboxylate, any phenolic group is there that is deprotonated in the physical condition, then we can have such kind of the interactions. So recent survey revealed that there are different kind of the enzymes which can allow such kind of the interactions. Like if we talk about enzymes like hydrolase, transferase, which involve such kind of the type 1 type of the interaction. But in the case of this graph, we see mainly transferase and again hydrolase, which are involving this second type of the interaction where lysine arginine working as a donor of salt-rich interaction. So analysis indicated that drug bank has got around 10,000 molecules and protein data bank have around 30,000 molecules. They identified that to make such kind of the interaction, we should have the anion, right? So they found that most of the whatever the ligand present in the drug bank or in the protein data bank, we have 30,000 molecule across the 180,000 proteins, which are mostly having this carboxylate anion, other phosphate related or sulfate related ligands, which can make such kind of the interaction. But most of the cases, in 66%, we have the carboxylate type of the molecules. But if we see protein data bank, we have around 70% of molecules which has got this type of the anion. And to make this second type of the interaction, we must have aspartate and glutamate, and the ligand should have the protonated amine. So amine may be anything, primary amine, secondary amine. So data reveals that in the protein data bank, we have around 54% of molecules which has got a secondary amine. We have also examples for primary, secondary amine and other imidazole-based molecules. So this is a representation what would be the distance and what would be the angle to make such kind of the interaction. This is the aspartate, this is the glutamate. So here we see on the x-axis we have the distances. So we in the most of the cases, so these chart are for, this is the for primary amine, this second chart is for secondary amine, and this is for tertiary amine. So in the most of the cases, what we see, the distance is around 3.5 between the hydrogen and the oxygen. And if we see angle, is quite varying, but most strongest type of the interaction are made if we have the angle around more than 150, we can say, because high density is here. And this black dot is indicating that this example present over here for each cases. This is example where lysine or arginine, any basic residue is present in the protonated form, and if it is interacting with any anion, here this is example where it is interacting with the carboxylate anion, and you can see again, and these have mostly distance around 3.3 angstrom. Again, they are highly populated for angle more than 150. So GPCR, there one database is there, and there a lot of mutational data is present. So they have identified that there is an interaction between the protonated anion protonated species with the any acidic center. Okay, if we replace, because such kind of the interaction are because of the formation of a salt-rich interaction. If we replace that aspartic acid or glutamic acid present in the active site, especially I think, yeah, aspartate is quite common in the most of the GPCR and the TM3, transmembrane 3. So if we replace that aspartate by alanine, there is a decrease in the activity 126-fold. Okay, if we replace that aspartate by aspargine, which has got a amide group, there is again decrease in the activity 63-fold. And if we replace that by similar molecule which has got a one additional carbon, glutamic acid, there is again decrease in the activity, though it has got a acid group, because there is additional carbon is there. There is no proper fitting between this cationic center and anionic center. That's why there is a decrease in the activity. So I think more than 100 examples are there where we have this mutational data for such kind of the interaction in the GPCR. So this is the drug where is showing how this aspartic acid interacting with the protonated amine. Another kind of the interaction is the van der Waals interaction. These are the very weaker kind of the interaction. So if molecule has got a electron density, if the perturbation is there, that lead to the formation of the temporary dipole, and those temporary dipole lead to the formation of the plus minus centers, and that lead to the attraction between the opposite dipoles. So to make such kind of the interaction, two species should be in the close contact. And if we see what is the energy contribution, what is the stability of such kind of the interaction, it is very, very less. It accounts around 0.5 kilocalorie per mole. It is very difficult to fine tune such kind of the interaction in the drug discovery or medicinal chemistry. Another interaction, the ion dipole or dipole-dipole interactions, as their name indicating that one ion is interacting with dipole. So here we have the permanent ion and that interact with other dipole molecule, which has got a positive negative centers. Another procedure that we can have one dipole, it can induce the dipole, and other molecule either we can have the two independent dipole that lead to such kind of the interactions. So whenever you put whenever you put any salt molecule in the water, so what kind of the interaction can happen there? So as we know that salt molecule NaCl, it has got an ionic interaction, okay, between the chloride ion and sodium ion. But when you dissolve it in water, there is a ion dipole interaction. This is the sodium ion, this is interacting with different water molecule. This is the chloride ion interacting with the permanent dipole of the water molecule. So similarly, there is a possibility that if we have any permanent dipole in the ligand molecule, and as we know that we have permanent dipole is there, like aspartic acid is there, it may interact with the carbonyl group. And similarly, if we have any like lysine is there that present in the protonated form, if arginine is there, it can also interact with the permanent dipole. So such kind of the possibilities are there. Here is one example. You can see here it has got a two type of the interactions. So in this side you see the dipole is there. It is making ion dipole interaction with the acidic group, and in this side here the cyano group or nitrile group is there, which is interacting with the hydroxyl group by the dipole-dipole interaction. So if we compare dipole-dipole interaction with the ion dipole interaction is much stronger compared to the dipole-dipole interaction, because in the case of ion dipole we have more electrostatic interaction between the interacting species. Next is the hydrogen hydrogen bonding interaction. And this is quite popular interaction. People try to optimize, improve this kind of the interaction to get a better affinity. So in this concept, we have the two electronegative center. One of the electronegative center has got a hydrogen. So electronegative center which has got a hydrogen, we call it a hydrogen bond donor. And another electronegative center which do not have any hydrogen, we call it a hydrogen bond acceptor. But because this hydrogen is attached to the electronegative center, there is a shift of the electron density of the sigma bond toward the electronegative centers. They lead to the creation of a positive center across this hydrogen. As we know that this hydrogen has got only single electron and it is departed toward the electronegative center. Now this nucleus is almost exposed to this electron center or lone pairs of the another electronegative center. Therefore, there is a such kind of the interaction is there. So to have a such kind of the interaction, we should have the two species, hydrogen bond donor or hydrogen bond acceptor. So another term we can say we should have the Lewis acid and Lewis base. So usually these kind of the interaction play very important role for the recognition process, either interaction between any specific ligand or protein or interaction between antigen and antibody. So before any these inhibitors or protein is interacting, because these are highly solvated. So in order to make such kind of interaction, there is a need of a desolvation of these reactive groups. Then such kind of the interactions are possible. So if we see on the energy level, energy is quite varying. So there are examples which involve very less amount of energy. There are also examples which involves stabilization by 40 kilocalorie per mole. So in this table, I have listed the distance between the hydrogen bond acceptor and donor. So in this first quadrant, you see this hydroxyl group is involved in the formation of the hydrogen bond, and distances are from usually 2.6 to the around 2.8. But if we see if an NH is involving in such kind of interaction, distances are from 2.9 to the 3.5. These are somewhat higher compared to these examples. But in opposite case, if the oxygen is involving or is working as a hydrogen bond acceptor, then again distances are somewhere from 2.8 to 3. But if the nitrogen center is involving such kind of the interaction, then distances are 2.8 to 3. So for the, if NS center is there, there is a more distance between the interacting species. If hydroxyl group is there, there is a somewhat less distance between the interacting species. So because this hydrogen bond is directional, because there is involvement of the lone pair of the one of the electron negative center or hydrogen bond acceptor. If we compare these two configurations over here is on, so this is the strong interaction, this is the weak interaction, because this orientation gives the direct overlap of lone pair of this oxygen with the hydrogen. But in this, here we see the lone pair are in this direction. Here there is no direct overlap, so therefore this is a weak interaction, this is a strong interaction. So because one molecule can participate in the multiple kind of the hydrogen bonding interactions, so like here one species involving with two other species, so we can say this is a bifurcated hydrogen bond or a three-centered hydrogen bond. So people have classified the hydrogen bond in the different category based on the amount of energy released in this kind of the interactions. Sometimes people call strong, medium, and weak. Sometimes very strong, weak. But based on distance angle, we can easily differentiate between this type of the interaction. Like for distance between the electronegative center is from 2.2 to the 2.5 angstrom. This is the strongest hydrogen bond. For the strong, we can have the distance from 2.5 to the 3.2 angstrom. For weak interaction, we can have from 3 to 4 angstrom. And if we talk about the angle which is required for such kind of interaction, if it is close to the 180, we can say, okay, this is very, very strong bond. And if we somewhere from 130 to 180, we can say, okay, is a moderate strength. And if it is varying from 90 to 180, because strength also depends on what kind of the functional groups are there, then we can say it is a weak interaction. Another parameter is that because this very, very strong hydrogen bond has got a pronounced covalent character. But if we see these two, this has got a weak covalent character and there is no any covalent character. So to make a hydrogen bond between interacting species, if we have any species which has a higher electron density, then we can say, okay, it can work as a very good hydrogen bond acceptor. So if we have any anionic species, of course, it has more electron density, so it is working, it will behave as a good hydrogen bond acceptor. These are the other examples which do not have charge. We can say these are the moderate hydrogen bond acceptor, and these are very weak. Similarly, if there is a chance that lone pair of the electronegative center involved in the delocalization or resonance, then because those electrons are not available, so we can say that is the formation of weak hydrogen bond. So tertiary amine, because this is not involving in any kind of the delocalization, so electrons are available, so it can form a good hydrogen bond. But if we see these examples which are involved in the delocalization of the electrons, so we can say these are the weak hydrogen bond acceptors. Similarly, if we compare the hydrogen bond donors, if any molecule have the more electron deficient hydrogen, of course it will make a very strong hydrogen bond. So if we compare these two, this will make a strong hydrogen bond because there is more this hydrogen more electron deficient compared to this hydrogen. So here is one example. If you see these two structure number 3 and 4, both are almost similar, just these are varying by just presence of the NH3 plus. Okay, so this addition of the only one substitution here lead to the increase in the activity by 500-fold, because this new addition of this functional group lead to the formation of the new interaction, and you can see there is huge change in just replacing only one functional group. So based on the what kind of the hydrogen bonding is, we can say, okay, there is a possibility of the two type of the interaction. It may be intramolecular, it may be intermolecular interaction. So if only atoms of only one molecule are involving, we can call them intramolecular. If two species are involved, we can say it is a intermolecular interaction. But whenever there is involvement of the intramolecular hydrogen bonding, we can say this gives the rigidity to the molecule. In addition, because these atoms are not free to participate to the surrounding environment, so this lead to the increase in the lipophilicity as well as increase in the membrane permeability, but at the same time this also lead to decrease in the solubility of a molecule. So here is an example. This is a very simple molecule, and here we have the carbonyl group, and here at the nitrogen has got a methyl group. So solubility of this molecule is very good. And if we replace this methyl group by hydrogen, here in this example 3b, okay, this lead to the formation of 6-membered hydrogen bond, and that lead to the decrease in the solubility as well as increase in the log D value for this molecule. So this way people are fine-tuning to optimize their molecules, how to fine-tune to increase the lipophilicity or solubility. This formation of intramolecular hydrogen bond can also affect the pharmacological action. There is one example. These two structures are the isomer of each other. If these substances are present ortho position, that lead to the formation of the intramolecular hydrogen bond. And if these two substances are present at the para position, then there is no possibility of formation of any intramolecular hydrogen bond. So this molecule is a very good antibacterial agent compared to this. Next is the hydrophobic interactions. These are very important interaction, and these are associated interaction between lipid-like molecule with the aqueous environment. So if you put any oil drop in the water molecule, that lead to the increase in the entropy of a system, because that hydrocarbon molecule, that oil drop, is not able to dissolve or mix with the water molecule, and therefore hydrocarbon have tendency to associate themselves. Similarly, water molecule also try to reorganize themselves. So such kind of the interaction lead to the such kind of means increase in the interaction for the similar kind of the molecules. Hydrophobic molecule try to make interaction with the another hydrophobic molecule, and polar molecule like water molecule, they try to make interaction with another water molecule. That lead to the such kind of the interaction. Suppose here we have the one drop. Whenever we add one drop of the oil, that lead to the increase in the entropy of the surrounding water molecules. If we add another molecule, that lead to the similar behavior. Now the tendency that these oil molecule will try to connect each other. They will try to reduce their surface area. Similarly, this water molecule try to come together. So this lead to the increase in the activity, and sometimes such kind of the interaction are sometimes very, very beneficial. If we talk about any protein and ligand, because before they make any such kind of the interaction, these are highly solvated. To make interaction, those water molecules should be replaced. Okay. So here is one example, 1 and 2. So these are look like very similar. And in the second molecule, just there is a replacement of the methyl group, and that methyl group replace the unfavorable water molecule present in the shallow cavity, and that lead to the increase in the activity for such this molecule. You see here activity was 341 nanomolar, now it is 7 nanomolar, is highly active. This is called magic methyl effect, but this is not true in every cases. But there are several examples. Just by fine tuning methyl group, it may lead to the drastic change in the activity. This is the variant of the hydrophobic interaction. We call it pi-pi interaction. If we have the 2 pi system, they can interact. So how they can interact? There are 3 possibilities are there, like T-safe, in which this is the orientation, and this is slashed or parallel displaced, and next is sandwich, in which the 2 rings are completely parallel. If we compare their stability, this middle one is the most stable, because here we can see the positive center is interacting with the negative center. But in this case, here positives interact with positives, so this is somewhat less stable. So such kind of interactions are quite common in the like DNA, RNA, where we have the several bases which are connected by such pi stacking. So there is one example. These are two molecules. This molecule number 5, this is one of the inhibitor for this hymen soluble epoxide hydrolase. So this has got around 700 nanomolar activity. And there was one cavity. If you can put some hydrophobic groups, so by placing this phenyl ring, it lead to the fitting of this phenyl ring in the pocket, which is surrounded by several aromatic ring containing residue that lead to the increase in the activity because of the pi interactions. There is a possibility of formation of the amide pi stacking interaction, and these kind of interactions are quite common where there is a amide bond of the backbone of the protein is involving with several heterocyclic system. So here you can see these are 3 examples. As we are increasing the dipole moment of these 3 rings, there is increase in the stabilized energy of a ligand. Anionic pi interaction. This is the case where one anion is interacting with the pi system. We know that pi system is also rich in electron density. How we can make such kind of interaction? Such kind of interactions are possible if the ring is the electron deficient. If we put any electron withdrawing substituent over the ring, then we can make such kind of the interaction. So such kind of interactions are not common in the biological system because whatever the rings we have the molecular system like phenylalanine, tyrosine, tryptophan, which are electron rich. So we do not have such possibility. So here is one example. This is one at porphyrin type system which has got a 4 pyrrole ring in the center, and in the wall here we can have the different heterocyclic system. So there is a possibility that if we add any anion, it can have the 2 type of the interactions. Okay, in one type just it making hydrogen bonding interaction with those 4 different pyrrole rings, and another possibility that those aromatic systems can also involve in the formation of a hydrogen bond. So here is the example for fluoride. Suppose if we add the fluoride to the system, this fluoride will make 4 intermolecular hydrogen bond with these pyrroles. In addition to that, it will also make anion pi interaction. So case where we have the such kind of the interaction, anion pi interaction as well as hydrogen bonding interaction, then we have the around complexation energy 7.6 kilocalorie per mole. And if we do not make interaction with this pi system, then we have the interaction energy 67 kilocalorie per mole. Similarly, for the chloride, if we have the all type of the interaction, we have the complexation energy around 48 kilocalorie per mole. And if we do not make an interaction with the chloride and just only hydrogen bonds are there, then we have the stabilized energy around 41 kilocalorie per mole. So if we compare the fluoride ion is a very good anion to make a anion pi interaction compared to the other halogens. Cation pi interactions. If any cationic species involving interaction with the pi system, we can have such kind of interaction. So we have some of the residue which can make the cation, or which can present the cationic form in the physiological condition, like basic residue like lysine, histidine, arginine. These can be present in the protonated form, and we have other residue like phenylalanine and tyrosine which can participate or can provide the pi system. So like that, here we can see this is the ligand, and which has got a amine group which is present in the protonated form. This is the indole from the one of the tryptophan residue, and we will make the such kind of the interaction. So people have explored a lot how to increase the strength such kind of interaction. What is the nature if we have the cationic species? What is the nature if we have the simple hydrogen bond donor, and what if we'll take the D metal cation. So if we have such kind of the simple alkyne and earth metals, cations, then we have mostly predominantly electrostatic interaction, and if we have such kind where neutral species are involving, there is a participation between orbitals, and if we have such kind of the interaction, those are the completely different what we see here. So there is one example. So these are 4 different molecules, and if we optimize the cationic species, okay, here you can see if only amine group is present, then activity was very less, and if we substitute methyl group to methyl group, if we make a quaternary ammonium, and you can see there is a increase in huge activity because of cation pi interaction. This cation is making interaction in the cavity which has got several aromatic centers. So I think this is the last interaction we are covering here. So this is the halogen bonding, halogen interaction. And if we see the geometry and distances are quite similar to what we have seen in the case of the hydrogen bonding interaction. So halogens has got a electrons, and if any halogen is polarized, iodine is more polarized compared to the fluoride. Fluoride at all is not polarized. We can't polarize it. So due to polarization, there is a creation of the electropositive centers, or we can say sigma holes, which are electron deficient, which are present at the tip of the halogens. So these are electron deficient centers, okay, and those electron deficient centers can take electrons from the electron rich or any heteroatom which can donate the lone pairs. That lead to the formation of such kind of the interaction. So here this any halogen may be here, but fluorine is not involving this kind of the interactions. There is no generation of the sigma hole in the case of the fluorine. And here we can have any kind of the heteroatom which can give the lone pairs. So if we talk about which halogen make the strongest type of the halogen bond, this is the iodine which can make the strongest, and strength of that halogen bond of iodine is almost similar to the hydrogen bond. And this iodine has got a largest sigma hole, therefore it is making the strongest interaction. So here is a comparison of the geometry of these 2 different bonds. Like as we know that in the case of the, to make a very good hydrogen bond, the distance should be around 2.2 to 2.5, and angle should be near to the 180. But to make this kind of the interaction, okay, the distance, this distance between halogen donor and halogen acceptor, it should be equal to greater than for whatever we have the any distance for covalent bond, but it should be less or equal to the sum of the van der Waals radius. So there are several examples in the literature where the people have optimized such kind of the interaction and lead to the formation of a very good, or they have identified very good inhibitors. So here is one example where they have identified bromine is working very good because of the good halogen bond formation with the tyrosine oxygen. So one recent survey, I think 2 years back, they have identified what kind of the interaction are present, because now currently protein data bank has got around 185,000 molecules of protein and around 30,000 different kind of the ligand. They have analyzed the whole library. They found that in the most of the cases, whatever the ligands present in the protein data bank, mostly we have the hydrophobic interaction followed by hydrogen bonding interactions. Then pi stacking interaction, then weak interaction where hydrogen from any carbon can interact with the oxygen of any carbonyl group or any other heteroatom. Then salt-bridge interaction and amide stacking interaction, and finally cation pi interaction. So as a medicinal chemist or as a drug designer, you can fine-tune these different kind of the interactions, and you can make drastic change to see is there an improvement or a drastic decrease in the activity. So with this discussion, I end my talk. Thank you everyone listening to me, and I would like to thank to my supervisors, my NIH postdoc supervisor Mark Nicholas, and this is software developer Paul from Germany. And this is the Professor Lilita Guru Prasad from School of Chemistry. I was working with her as a postdoctoral fellow. And Dr. P. Bhartam, he was my PhD supervisor. And Dr. Evans Coutinho from Molecular Pharmacy, he was my master supervisor. So I'm very thankful to them. Yeah, we can take if we have some questions.
Thank you, Dr. Dhakkad, for such a wonderful talk and taking time to explain the basics and how they can be used for drug design applications. So we have a question on the chat box. How we can choose what water molecules are important for interactions with water receptor interaction data when the data is not available? Can you read it? Chat box?
Now I got a question. So because we have around 1,85,000 of the crystal structure for protein. So because the resolution is quite varying, so it is very difficult to get a conserved water on that resolution. So now some tools are there. If by chance any protein at low resolution level someone has solved, then we can compare, okay, there is possibility to have a conserved water, then we can account for that. Otherwise, very difficult to involve that. So there are some examples. People have solved the structure where we have the conserved water, and those involving the interaction or working is a bridging between ligand and the protein.
Yes, I guess there is one more question on how we can reduce off-target toxicity at the time of designing covalent inhibitors.
How we can reduce the off-target selectivity for designing covalent inhibitors?
Off-target toxicity.
Yeah, so it is a very good question, because nowadays a lot of research is going on how to design and how to make a selective covalent inhibitor. As we know that there are several electrophilic warhead groups are there which can make covalent bond, and we don't know nature of off target. So you can fine tune by putting some bigger groups, okay, bigger groups, but because it is very difficult to fine tune the toxicity, because there are several targets are there. If you do n number of assay, if you come to know that okay, this molecule is also binding there, then you can fine tune relatively. Otherwise, it's very difficult.
Yes. So once again, I would like to thank Dr. Dhakkad for delivering this nice presentation. So with this, we come to an end to the first session of day 2. And in the afternoon, we will begin at 2:30 p.m. sharp. And our speaker will be Dr. Sam D. Weiser from University of Manchester. So we will resume the session then. Thank you.
Yeah, thank you.
Yes, we can go online. Yes, we are online, sir.
Okay, great. Good afternoon. Welcome back again to all the participants. Today we are delighted to have Dr. Sam D. Weiser as the next speaker. He works at the University of Manchester and he has done PhD from the University of Amsterdam. He is the Fellow of Royal Society of Chemistry, has won many awards, and he is a Ramsay Memorial Fellowship awardee. And he is also FRSC since 2011. And he basically works in multiple areas on biocatalysis, computational chemistry, reaction mechanisms, thermodynamics, physical chemistry, and he specializes in understanding spin state reactivity of multiple oxidoreductases and redox enzymes. I had the good fortune to visit his lab for a few months, and we did publish some good work from that. And today he is going to talk about second coordination sphere effects on enzymatic reactivity, insights from computation. So we look forward to that. Over to you, Sam.
Okay,
Please
thank you very much, Vaibhav, for this nice introduction, and thanks also for getting me involved in this nice meeting. So hopefully in one of the future meetings I can come to Guwahati again and visit you, because I find conferences easier face to face rather than Zoom. But in the absence of being allowed to travel at the moment, it is a good alternative. So yes, we're working on enzymatic reaction mechanisms, and I'm going to show some examples today on how the second coordination sphere actually affects the structure, reactivity, selectivity, and various other properties in enzymes. So as Vaibhav already told you, I'm a computational chemist, and we do use various methods and techniques ranging from density functional theory to quantum mechanics, molecular mechanics. And the main focus is on three different streams of research, essentially. First of all, we try to understand nature, so we look at natural enzymes, like, for instance, the cytochrome P450, that catalyzes a range of drug metabolism reactions in the body. They also do biosynthesis reactions, and that's not only the P450s that do that. There are various enzymes, iron-containing enzymes, mainly that are involved in biosynthesis reactions. And we try to understand how nature does these biosynthesis reactions, in particular because natural reactions often are very selective or stereospecific, and we try to understand how that is guided by. Then, as it happens, I'm based in an Institute of Biotechnology, so we also try to apply our understanding of these natural enzymes and see if we can find some biotechnological applications of these systems. And examples of that we've been working on in recent years are the biosynthesis of biofuels from enzymes. So that's basically formation of hydrocarbons from whatever natural reactants there are. But we also recently looked at herbicide biodegradation. So certain plants actually can biodegrade the herbicides farmers spread on them, which is of course good for humans, because that means we don't take these herbicides in, and that's lesser toxicity for humans as well as animals that eat these plants. And finally, we also look at biomimetic models. So that's mostly work we do with the Sastry group in Guwahati, which are synthetic models of the enzyme active sites, which take a metal and its first coordination sphere, similar to what you see in enzymes, and react in a solvent solution like acetonitrile or toluene or ideally water. And we often see differences in that sort of reactivity of these biomimetic complexes with the enzymes. And at the bottom of this slide, I show the few examples of nice cover pages we got. Sometimes you can negotiate these in Chemistry European Journal. So we have here two examples. So the first one on the left is an example of a study on a drug metabolism reaction we did, and particularly it was related to anti-epilepsy drugs, which in the liver could react through the cytochrome P450 in two different ways, either a desaturation channel, that's the pathway on the left, or a hydroxylation channel, the pathway on the right. And we try to understand how is the bifurcation of these pathways affected by our reactant, our oxidant, but also our environment. And the example of the right is, as you can imagine, that's a biofuel example, where it's another cytochrome P450 enzyme which takes a long chain fatty acid, and instead of hydroxylating the fatty acid, actually what it does a decarboxylation, which gives you a hydrocarbon, which of course you can burn, and that gives you your biofuel. So to recap of what essentially we do is we are interested in organic reaction mechanisms. So most of the systems we work on have at least one transition metal, which is mostly iron, but we sometimes also look at manganese or even vanadium or related. So we look at the overall reaction mechanism of what's happening in this fast reaction process, either in the enzyme or in the biomimetic model. We try to find what is the rate determining step, what is the active species that does the catalysis, and can we characterize some of these short-lived species so that they can be identified with EPR, NMR, or infrared or whatever, to get more insight into this overall reaction. And then we try to do some biotechnology sometimes on these systems by engineering the system and modifying either our oxidant or our protein, and hopefully that improves the catalysis or changes selectivity, which is either regio or chemo or the stereo selectivity, and that's all through bioengineering of these proteins. So as I said, we do biomimetic models sometimes, as well as the enzymatic system. So here at the bottom there are 3 biomimetic models, mostly work we did with the Sastry group, for instance, a manganese side on peroxo or an iron oxo or another iron oxo, with various ligand structures that affect their properties and reactivity. And in addition, we look at enzymes, as you can see here on the right, with this another nice cover page in Chem European Journal, where we either extract the heart of the active site of the enzyme and use a cluster model to study the reaction mechanism, or we take the whole enzyme, like here in the top, where we take the whole protein with a solvent layer and use quantum mechanics, molecular mechanics to study the full reaction mechanism. So as I said, we are particularly interested in these bifurcation reactions because that gives some different reaction products, and how can we guide a reaction to a certain product. So we came across here an example of this, of a cytochrome P450. So this is a cytochrome P450 called a GCO-A, which I believe is a P450 you find in fungi. And what this particular P450 does, it takes lignin fragments, so it's not a complete lignin chain this P450 takes, but monomers of lignin, such as guaiacol or seringol, that sort of small monomers, and these small monomers can be activated in this particular P450, and this P450 GCO-A does that in a strange way. It gives 2 products, namely either hydroxylation of the methoxy group, or you get an oxidative ring closure to form this acetal structure. And essentially that's a desaturation. So we were wondering what are the pathways that lead to these 2 products, and is the substrate binding and positioning in particular relevant to this. So when we look at the active site, as I mentioned, this is cytochrome P450, which has a heme group, which binds to a thiolate of a cysteine of the protein, and it attaches to the protein through this thiolate, as well as with some hydrogen bonding interactions of these propionate side chains. But when you look at the substrate binding pocket, actually it's quite apolar, with mostly aliphatic and aromatic residues, but also a few negatively charged residues like this carboxylate of this glutamic acid. But as you can imagine, the substrate can bind in many different orientations. And indeed, when we do a simple docking procedure, I think we used auto-dock in this case, we get several low energy conformations with like a draw here on the right in purple, with either the methoxy group pointing down towards the heme or the phenol as well as the methoxy groups pointing down towards the heme. So we said, let's do two sets of calculations with a very large cluster model. And in this case, we had a cluster model of 302 atoms, because as I said, the second coordination sphere sometimes is important, and that includes then the substrate binding and positioning. So we decided to take the heme and the substrate binding pocket and the substrate in either of these two conformations. And we wanted to see, can both conformations lead to the same products, or do we get different products in conformation 1 than in conformation 2? And as it happens, the calculations show that when the methoxy group points down and the phenol group points up, then actually we only get methoxy hydroxylation, whereas when the phenol as well as the methoxy group points down, then we get a desaturation, we split off a water molecule, and we get this acetal formation. And I've got some energy landscape to show how that works. So there's a lot of numbers, a lot of steps happening here. So we start with compound 1, which is an iron 4 octoheme, and the thing with this iron 4 octoheme is that it has two close-lying spin states. It's either a quartet or a duplet spin, and as you can see, we calculate those within 1 kcal per mole. They're sort of in equilibrium probably. And then we started with the substrate with both the phenol and the methoxy group pointing towards compound 1, and tested, can we abstract a hydrogen atom from the phenol, or can we abstract a hydrogen atom from the methoxy group? So as you can see, a hydrogen atom abstraction from the phenol group has a very small barrier of almost 1 kcal per mole or lower, which is of course not surprising because a phenol OH bond is very weak. So it's easy to abstract that hydrogen atom, and we relax our system down to an iron 4 oxo with a radical on the substrate. And then, of course, from this iron hydroxo with a phenol radical, we can't do an OH rebound because that would give an iron or an hydroperoxo seringol, which is of course not a stable product. So what happens is this iron 4 hydroxo picks up another hydrogen atom from one of these methoxy groups, which now has a small barrier, and it will lead to a water molecule and this biradical with a radical on the methoxy, yes, and a radical on the phenol. And of course, these two radicals pair up to close the ring very quickly to form the acetal product. So the alternative pathway is a hydrogen atom abstraction from the methoxy group, which, as you can see, has quite high barriers, 13 to 17 kcal per mole, leading to also an iron 4 hydroxo with a radical, but that's much less stable as the radical on the phenol, as you can see, by 10 kcal per mole. Nevertheless, if that can be formed, then this radical intermediate 2 here, M1HAB, picks up a hydrogen atom from the phenol very quickly, and then also gives this biradical and leads to acetal products. So as you can see, when the phenol and the methoxy group both point towards compound 1 and are essentially within hydrogen bonding distance of the oxygen atom of compound 1, then the most likely reaction pathway is desaturation to form the acetal products. So what happens now when we have the substrate in an alternative orientation? So we take now the substrate with the phenol group pointing up and the methoxy group pointing down. And now what we see is we can only abstract the hydrogen atom from the methoxy because the phenol is hanging very far away. It will take a lot of energy, by the way, to move or reorient the substrate and abstract the hydrogen atom from the phenol, because it's quite tightly bound here with a lot of hydrogen bonding interactions, as I will show later. Nevertheless, what we see is we have small of quite high barriers for hydrogen atom abstraction now from this methoxy group, which leads to another radical intermediate, and now there is a rapid OH rebound to form the hydroxylated product inside. So as you can see, with the phenol far away from compound 1, we get methoxy hydroxylation, whereas when the two OH group and the methoxy group point toward compound 1, then we get the desaturation pathway. So here's the scheme that shows that how tightly the substrates are bound. So with the green arrows, I show the key hydrogen bonding interactions of the substrate with essentially the second coordination sphere, which is the substrate binding pocket. And you see here there's the carboxylic acid of this glue 249, hydrogen bonds with the methoxy group. There's an amine group here of this glycine residue, which forms a hydrogen bond with the phenolic oxygen, and the valine carbonyl group is also forming a hydrogen bond with that phenol OH group. So as you can see, it's quite strongly bound in this substrate binding pocket. So tumbling over and bringing the OH group nearby is not a likely channel, and it is most likely that we just get CH3 activation and hydroxylation of that methoxy group. And here, on the right here, I show that in the alternative substrate binding orientation, it's also quite tightly bound. You again see here an amine group of this valine 241, which forms a hydrogen bonding interaction here with the phenol. There's a threonine alcohol group which nicely hydrogen bonds as well with the methoxy group. So overall, the substrate is quite tightly bound, and through all these hydrogen bonding interactions of the protein chains and protein residues in the substrate binding pocket. So overall, this work on this lignin biodegrading P450 enzymes showed that seringol, that was that substrate we used, can be activated in various different ways. And we see that different substrate binding orientations and different substrate binding poses actually give different product distributions. So if the phenol group is close to compound 1, then the most likely channel is initial hydrogen atom abstraction from the phenol, because that's simply the weakest OH bond, and that hydrogen is the easiest to abstract. But then the subsequent reaction will be another hydrogen atom abstraction and desaturation rather than an OH rebound type reaction. So that's the acetal pathway that only can happen after an initial phenol hydrogen atom abstraction. And if the phenol group is pointing away from the heme, then you get the alternative methoxy hydroxylation pathway. Okay, so this first study shows that actually the second coordination sphere is very important because it positions the substrate, it orients the substrate, it binds it tightly and locks it in a specific orientation. But of course there are more interactions that can influence the substrate. So it can be with aromatic groups or aliphatic groups and steric interactions that lock it in position. But as I showed, there can also be hydrogen bonding interactions. These hydrogen bonding interactions can be with a substrate, they can be with a ligand bound to the metal center. It can be an equatorial ligand or an axial ligand. So these hydrogen bonding interactions towards your ligand of the metal can actually change the chemical properties of the metal center and even the redox potentials and that sort of things. And you can even think there may be even salt bridges are either towards a ligand of the metal or to the substrate if it has a carboxylate group. And finally, there can be positive charges in our substring binding pocket or in the second coordination sphere. And these long-range charge-charge interactions or these charge-dipole interactions, they can all influence the structure, reactivity, and the properties of this metal center. So obviously, when you set up a model of an enzyme, you need to take all of those sort of effects into consideration. So if you ignore these charges or these salt bridges or key hydrogen bonding interactions, then you may weaken your model and may get completely the wrong results. And I'm going to show some examples of that in a moment. So here's one of the oldest examples I could find from work I was involved in, where we looked at second coordination sphere effect. And this is some quite old results from 2002, 20 years old now, where we took compound 1, a minimal cluster model, just a bare heme without side chains with thiolate as the actual ligand. And then we studied the reaction with a very simple molecule, propene, and either studied the double bond activation or the hydroxylation of the CH3 group. And we did that on, of course, the quartet and duplet spin states. And when you do that reaction in the gas phase with a minimal cluster model, then what you find is these hydrogen atom abstraction and epoxidation barriers all within a very narrow window of half a kcal per mole, which is not in agreement with experiment, because the J. Groves group did experiments on cytochrome P450 with propene, and they found methyl hydroxylation as dominant pathway. So the gas phase results clearly give the wrong products. So what we then did is we said maybe we need hydrogen bonding interactions towards this thiolate, because this thiolate has a push effect towards the heme, donates electron density, and the charges on the thiolate will be influenced if there is a hydrogen bonding interaction there, and also the push effect. And indeed what you see when you add these two simple ammonia molecules in the model to mimic these hydrogen bonding interactions of protein chains that are there in the binding pockets, then what you see is the hydrogen atom abstraction barriers are stabilized, whereas the epoxidation barriers go up. So we're already starting to see the right pattern in selectivity, and we get even a better selectivity if we add a polarized continuum model with a dielectric constant of, let's say, 5.7. That gives a bit more realistic dielectric continuum as should be in an enzyme active site. And as you see now, we reproduce the experimental ordering of the transition states. Together with Dr. Dixit from NIPER, we had a paper, as he mentioned, where we looked at redox potentials of heme systems. And Dr. Dixit showed that if you've got positively charged ions essentially in the second coordination sphere, and then the redox potential of the heme is already affected. And even if these charges are 10, 15 angstrom away from this iron center, they still have significant effect, that it does affect redox potentials and therefore electron transfer rates in the P450 catalytic cycles. So here's another example that actually when you see these cations in your second coordination sphere, it's actually essential to keep them in the model, because otherwise your energetics will be completely wrong and not match experiment. I'm not going to give more details on the Dr. Dixit's work. I'm sure he will give some talk himself later on. So I want to show one more example where we did extensive testing on models and compared our system to experimental data. So an extremely well studied non-heme iron system is taurine alpha-ketoglutarate deoxygenase. It is an enzyme involved in the cysteine metabolism in the body. So in the brain, cysteine needs to be metabolized because sulfur residues with metal ions give plaques in the brain, and that can lead to Alzheimer's and Parkinson's diseases and so on. And so the body has mechanisms to get rid of sulfur-containing compounds. And the oxidation of taurine is one of the steps in this mechanism. And what the body does, it selectively puts an OH group on the C1 position of taurine, which then leads to removal of sulfate from the molecule and further degradation of the molecule. So as I said, it's been studied extensively by the Bollinger group 10, 20 years ago, as well as the Hausinger groups, and that they've characterized the iron 4 oxo species with the infrared, EPR, Musbauer, and various other techniques. And they established a rate constant for hydrogen atom abstraction from taurine of 13 seconds minus 1 at 5 degrees centigrade. So we thought, let's set up some models of this tau-D and see if we can reproduce that rate constant and what affects this rate constant. And in addition, what we do is we try to abstract each of the 4 hydrogen atoms of taurine. So the 2 hydrogen atoms of C1 and the 2 hydrogen atoms of C2. And we want to see if more than 1 hydrogen atom can be abstracted by this enzyme, or if they are equal in energy. Okay, so we set up several models. So we started with a minimal cluster model here, which I label model A, which is simply the iron oxo with the first coordination sphere of residues, which are 2 histidines and 2 carboxylates, one from an aspartate and one from a succinate, and the substrate taurine. That's only 72 atoms. Then we expanded model A with the substrate binding pocket of taurine, which, as you can see, has all these aromatic residues here. There's a tyrosine also. There's also a histidine here. But we didn't know exactly what the protonation state of that histidine needed to be. So we started off with a neutral histidine in model B to get 244 atoms. There's also an arginine here with a positive charge and a few hydrogen bonding residues on this end here, asparagine. And then we expanded it even further and decided to put now a proton on this histidine 70. And we had a few more residues here on the left that give more hydrogen bonding interactions here to the amine group of the substrate. And that's our third model of 279 atoms. And then in addition to these 3 DFT cluster models, we also did QMMM. So we did QMMM with a QM region, which essentially is model A. We did QMMM with QM region of essentially model B, as a comparison to the DFT cluster models. So I'm not going to show all the results. I'm just going to show this table where I have the hydrogen atom abstraction, free energies, so either from the C1 position or the C2 position. So as I told you, there are 2 hydrogen atoms on C1 and 2 on C2. So I only have here the barrier, the lowest energy barrier of those 2. And as I said, there's an experimental rate constant, which you can convert to a free energy of activation using transition state theory, which gives you then a value of 14.8 kcal per mole. So as you can see, when we have the DFT, the minimal DFT cluster model of just the first coordination sphere of the iron center and the substrate, then we get a terrible free energy of activation, which is 10 kcal per mole off from the experimental data. So the minimal cluster model clearly is not a very good model. So that's not really surprising because it doesn't have the substrate binding pocket in there. However, it does give the right selectivity because it gives C1 hydrogen atom abstraction below C2 hydrogen atom abstraction. So there is something positive about the small cluster model. So if we then interestingly take QMMM and use the minimal cluster model, just the first coordination sphere and the substrate as the QM region, as you can see, the energetics don't improve a lot. So we still get a very low barrier of 6.7 kcal per mole of hydrogen atom abstraction from C1. C2 is still very, very high. So in both cases, either a cluster model or minimal cluster model or QMMM with a minimal QM region give very poor energetics, poor kinetics, but do seem to give the right selectivity. If we expand it to cluster model B, which has the whole substrate binding pocket in it, but has the histidine deprotonated, then as you can see, we get quite a good agreement of the C1 hydrogen atom abstraction with experiment. It seems to be within 1 kcal per mole. But unfortunately, I get quite a slightly lower barrier for the C2 hydrogen atom abstraction. So that means that the large cluster model B, which has the substrate binding pocket included, gives reasonable energetics of C1 and the right kinetics, but it doesn't give the right selectivity because there's another barrier suddenly lower in energy that shouldn't be lower in energy. So we then expanded model B to model C, where we put a proton on the histidine. And as you can see, the C1 hydrogen atom abstraction is still okay. We are within 1.5 kcal per mole of the experiment, so that's quite a nice match, I would think. And the C2 hydrogen atom abstraction has gone up steeply to well over 20. So with a large cluster model C, where we have the full substrate binding pocket and presumably the correct protonation states of all the residues, we now have the correct kinetics, the correct energetics, and also the correct selectivity. So we also did QMMM with model B, and it still doesn't give the right thing. So in our case, as you can see, QMMM wasn't particularly a good method for tau D to calculate the right selectivity or the right energetics of the reaction. And the only thing that worked well is this large cluster model of 270 atoms that has all these hydrogen bonding interactions included. So to recap this table, the small cluster models or small QM region QMMM calculations give the correct selectivity but wrong kinetics, and only large cluster models with the His70 protonated give both good kinetics and the correct selectivity. And that's the minimal, apparently, that you need to reproduce tau D work. So we don't try to find out why are the energetics and the kinetics so different, and how come they're so sensitive to the protonation states of histidine residue. So we decided to just take the substrate, and this is the isolated substrate I'm now talking about, no protein around it, no iron around it, and we calculated the CH bond strength of each of these positions. So we simply took the substrate, did a calculation, then we removed the hydrogen atom, did a calculation, and we calculated the hydrogen by itself, and calculated the energy difference for that to get the bond dissociation free energies of all these positions. And as you can see in the gas phase, the C2 bond strength is 89.4 kcal per mole, whereas the C1H is 87.4 kcal per mole. So that means in the gas phase, you would get preferential C1 hydrogen atom abstraction over C2. And that's indeed what we see in the small cluster model, or even in the QMM with the small QM region, you get C1 hydrogen atom abstraction. So clearly we can perturb the system so that we can favorably abstract the C2 hydrogen atom abstraction. And to test that, what we did is we took the isolated substrate and applied a local electric field along the X, the Y, the Z, or the minus X, the minus Y, or the minus Z direction, and checked how is the bond dissociation energy changing. So if you do that field along the Z-axis, that's here on the right, you see the order C2 and C1 stay the same. The figure on the left is in the X direction, and it's more or less also here that the ratio X versus Y stays the same. But in the middle, you can see that when we have this applied electric field along the Y-axis, which is essentially along a CH bond, then you can see that if there's a field in one direction, I make the C1 bond the weakest, and if the field is in the opposite direction, I make the C2 bond the weakest. So I can actually play with my charge distributions around the substrate, and then favorable get C2 hydroxylation or C1 hydroxylation. That's essentially what this middle figure says. When a local electric field along the CH bonds of this substrate taurine can actually strengthen or weaken the individual CH bonds, either on C1 or C2. And by having this, probably having this protonated group of this histidine 70, this probably strengthens the C2H bond and weakens the C1H bond, and favorably then gets C1 hydroxylation with no C2 hydroxylation at all. So is that what we see when we look at the whole protein? So here I compare the dipole moment of the two cluster models we had, cluster model B with the His 70 deprotonated and cluster model C with His 70 protonated. And what you see is the dipole moment moves in a different direction when we go from model B to model C, because there's a positive charge missing, and some hydrogen bonding interactions are different, which affect our dipole moment and our charge distributions. So this big benchmark test set of calculations on taurine alpha-ketoglutarate dioxygenase was done using cluster models and quantum mechanics, molecular mechanics. We tried different size QM models. We tried QMM with different size QM regions, and we show here that actually the second coordination sphere is extremely important to reproduce experimental results, and particularly to get the kinetics and the selectivity in the right ballpark. So when you use a minimal cluster model or QMM with a small QM region for this particular system, that's highly sensitive to second coordination sphere effect, we get the wrong kinetics and energetics, and therefore it gives meaningless results. However, when we use a large model with a histidine protonated, then we get the right kinetics, we get the right selectivity, and we have a good representation of the actual enzyme. Okay, so let me give me another example of second coordination sphere effect in these non-heme iron enzymes. So we recently also worked on this enzyme, which looks very similar to tau-D, to be honest. It's called 2-trimethyl ammonio-ethyl phosphonate dioxygenase, which I abbreviate to TAMP-A. It has a substrate that looks very much like taurine, but has a phosphate group rather than a sulfate group, and not an NH3 group, but ammonio group. And this system reacts similar to tau-D, highly selective, and it puts again on the 1 position an OH group, not on the 2 position, and it also doesn't hydroxylate the methyl group of the ammonia groups. So we wanted to understand what in this case is the selectivity about, and if it would be similar to tau D, that there are second coordination sphere effects that influence the positioning and the selectivity. So this enzyme is this particular enzyme is not in humans; it's only in bacteria. But there is a related enzyme in humans that is involved in the carnitine biosynthesis. So there is an analogous enzyme to TAMP-A in humans. I think it's called BBO or something like that. Anyhow, we set up a cluster model. So this is the cluster model of the TAMP-A based on the crystal structure coordinates. As you can see, it's again an iron-4 oxo with binds 2 histidines and 2 carboxylates, 1 of succinate. In this case, we took a few protein chains surrounding the substrate binding pocket with all these hydrogen bonding interactions of these tyrosines. There's arginine, there's an asparagine here, and a few more residues that hold everything nice and tight together. So when we then do the calculations on the hydrogen atom abstraction from the C1 position, the C2 position, and the CH group of the methyl group, then we see is the lowest energy barrier we get of 11 kcal per mole is hydrogen atom abstraction from C1. So that's good news, because that's what the experimentalists say is happening in the real system. So we get the right selectivity directly. There is no rate constant, so we can't compare the number. The methyl hydrogen atom abstraction is a bit higher in energy, by about 4, and the C2 hydrogen abstraction is even higher than that. It's about 8 kcal per mole higher. So it's clear it's favorable C1 hydrogen atom abstraction. So here again, we took the isolated substrates, and in the gas phase, we calculated what the CH bond strengths are. And here what we find is that in the gas phase, the C2H is the weakest of the two at 84.9, whereas the C1 is much stronger at 89. So that means that in the gas phase, you would expect C2 hydroxylation rather than C1 hydroxylation, which obviously doesn't happen in this particular enzyme. So we reasoned again, probably there's an electric field here, or polar residues that change the CH bond strength in the model. And indeed, when you put a field along the Y direction, which is again along the CH bond strength, then in the positive direction, you weaken the C2H bond, whereas in the negative direction, you weaken the C1H bond. So a negative electric field along this one of the CH bonds should favor C1 hydrogen atom abstraction. And this is again, this is an electric field plot, how you gradually see that in one direction you weaken one bond, and in the other direction you strengthen that particular bond. Okay, so these electric field effects or dipole moments have the same effect, of course. They can favor C1 hydroxylation over C2 hydroxylation, and we expect that to be in play in this particular enzyme. And indeed, the dipole moment points along the CH bond of the model, and indeed, there seems to be a hint that dipole moment is important to guide the reaction to C1 hydroxylation. Also, when you do an electrostatic plot on the full enzyme, so this is the full enzyme structure, the full PDB structure, you can see a lot of red regions. So red is a negative charge, blue is a positive charge. So there's clearly in this protein a lot of negative charges and a lot of charge interactions and dipole moment that must influence the properties and the kinetics of this particular system in agreement with the cluster model that we calculated. Okay, so to summarize this TAMP-A work, so we did DFT studies on this TAMP-A enzyme, which is a non-heme iron enzyme that selectively hydroxylates the substrate. And again, we have a relatively large cluster model of 240 atoms. That includes first and second coordination sphere of the oxidant as well as the substrate, and ideally the whole substrate binding pocket. So the calculations are in good agreement with experiment. We find the correct lowest energy barrier. However, when we just look at the substrate in its isolation, then actually it would predict a different selectivity than what we can see, and we see that these electric field effects and electric dipole moments in the second coordination sphere actually influence the CH bond strengths and guide the enzyme to the specificity and selectivity that is needed for this particular system. Okay, so last non-heme iron example that I'm going to give is cysteine dioxygenase versus mercaptopropionate dioxygenase. Cysteine dioxygenase I already mentioned earlier. It needs to be detoxified in the brain, because otherwise it can lead to Alzheimer's or Parkinson's diseases. And cysteine dioxygenase catalyzes the first reaction in that process in the brain, where it takes cysteinate residues and sticks to oxygen atoms, essentially on the sulfur atom, to get cysteine sulfinic acid. So mercaptopropionate dioxygenase does a similar reaction. So this is not a human enzyme, but in bacteria where this happens. But a similar reaction is catalyzed. Two oxygen atoms are added to the sulfur atom of this system. Okay, so when you look at the crystal structure coordinates, so here are the crystal structure coordinates of the two enzymes. So the left is of cysteine dioxygenase, which has substrate bind. It doesn't have oxygen in it. It has an iron center, which is bound to 3 histidine residues, no carboxylic acid in this case. It binds the substrate as a bidentate ligand through the sulfur and the amine groups, and the carboxylate of cysteine is involved in a salt bridge with arginine 60 and forms hydrogen bonding interactions with this tyrosine and this histidine in the substrate binding pocket. And finally, there's this odd thing in the substrate binding pocket of cysteine dioxygenase, which is this covalent linkage between C93 and tyrosine 157, where on the ortho position you see a sulfur carbon bond where it's connected. I think there are only 2 enzymes in nature that have that sort of linkages. So it's also not understood what the function of that linkage is, and it maybe have to do with positioning the substrate and locking it in. But that's all hypothesis. For mercaptopropionate dioxygenase, there's a PDB, but it doesn't have substrate in it. So the purple the substrate which I have in here, we docked in ourselves. So this enzyme also has an iron. It also binds through 3 histidine, similar to CDO. So that's all very similar. There's also this tyrosine and histidine in the substrate binding pocket, but as you can see, the tyrosine now is not linked to cysteine, so it's more can move a bit more freely. The difference is that in the position of this arginine 60, there's now a glutamine, sorry. So there's no arginine there. And of course, the reason for that is that the carboxylate of the substrate is not pointing in that direction, because the carboxylate is proposed to bind directly to iron, and therefore the arginine you see in MDO is pointing behind the binding pocket you see here in the back arc 168. So that's the major difference between the two enzymes, that this arginine is in a different position and the substrate binds slightly different. It binds to the sulfur in both cases, but in MDO it's the carboxylate probably that binds to the iron, and in the CDO the amine group binds to the iron. and it misses also this linkage between tyrosine and cysteine. Okay, so many years ago, together with the Devish Kumar group in Lucknow, I think this was before we moved to Lucknow, we did some QMMM calculations on cysteine deoxygenase. And before that, I think in 2007, we did a DFT cluster model of about 110, 120 atoms. These two studies gave more or less the same results and found it's in a consecutive oxygen atom transfer mechanism where you start from an iron superoxo, then the oxygen atom attacks the sulfur to form this ring structure, then you get OO bond cleavage to form a sulfoxide and followed by a final oxygen atom transfer to get cysteine sulfinic acid products. And the calculations show that we start from an open shell singlet spin state with a radical on the oxygen and a radical on the iron, antiferromagnetically coupled. But then along this oxygen attack on sulfur pathway, we get a spin state crossing to a quintet spin state where the rest of the mechanism takes place. And that is the rate determining step in the reaction. So what we decided to do is we wanted to do cluster models because we had some good experience with these cluster models based on the Tau-D and the Tambay results that I already showed you. And in this case, we decided to, we said, well, we did a cluster model in 2007 of about 110, 120 atoms. So we said we need to go beyond that. So we chose a cluster model of 308 atoms of CDO. And basically to benchmark our calculations against the QMMM and the previous cluster models and see are there disastrous differences when we use a cluster model. And as you see, the cluster model uses the metal with the substrate and dioxygen, a large chain starting from set 184 all the way here until leucine 95. So that's a nice chain circling around the substrate binding pocket and includes two of the ligands of the metal and a few more chains that determine the substrate binding pocket and give all the hydrogen bonding interactions in that pocket. And we did the same for the MDO, that's the structure here on the right. That's now 324 atoms. As I said, it doesn't have this arginine 60 here, so we didn't include that one. The arginine is somewhere in the back. Here it is, 168. But we use a similar focus and active site structure as we see with the CDO. Okay, so here's an abbreviated energy landscape for the CDO using the cluster model. So in agreement with the QMMM, we find a singlet spin reactant iron superoxo. We get a spin state change from singlet to quintet with a transition state of close to 10. Leading to this ring intermediate, which then after a few steps collapses to this iron 4-oxo and sulfoxide, and then there's a final oxygen atom transfer to form cysteine sulfinic acid products. So most of these steps are very similar. The energetics is very similar to the QMMM. The only difference is that the second oxygen atom transfer barriers are a little bit larger with the cluster model than they were with the QMMM. So they are now almost competitive with this first oxygen atom transfer reaction. Then we looked at MDO, and it's not disastrously different. We start with a singlet, open shell singlet reactant. However, the quintet is actually very close in energy, within 1 kcal per mole, and it means that the quintet barrier is very low in energy in this particular case. Nevertheless, we still need to cross over from this singlet to the quintet, so that the spin crossing barrier may be a bit higher than that. And then we collapse to this ring structure that's quite exothermic, as you can see. Then we react to the iron 4-oxo, and that's another very exothermic step. I believe this is 45 kcal below the reactant structure. structures, so there's quite an exothermicity for MDO to reach the iron 4 oxo. And here as well we have a barrier of I believe 8 to 10 kcal per mole for the second oxygen atom transfer, which is rate determining, of course, depending on how fast this spin crossing here is from singlet to quintet. So overall, the MDO reaction shows that both CDO and MDO are stepwise oxygen atom transfer reactions. Both start from an open shell singlet iron 3 superoxo. In both there's a spin crossover from singlet to quintet at the start. And in both cases, the oxygen atom transfer, the second one is relatively high in energy, and it seems to be competitive with the first oxygen attack on sulfur. And finally, we find that actually this iron 4 oxo is highly stable in MDO, and much more than it appears to be the case with CDO. So that was a bit unusual for us. We didn't expect that result. So we thought, let's analyze that a little bit more in detail. So on the right here, I have an overlay of the two structures, the iron 4 oxo complexes. So there's not much difference to see. It's just different protein residues that were included in the two models, but the first coordination sphere is more or less in the same place, so there's no disastrous differences we see down there. The only difference we see is the structure here on the left. So in the equatorial plane, the iron 4 oxo has two histidine ligands, a sulfoxide and a neutral amine. Whereas in MDO, the iron 4 oxo has still the two histidine ligands and a sulfoxide, but now it has a carboxylate as the fourth equatorial ligand. So there's an anionic fourth ligand rather than a neutral fourth ligand. And that's the difference between these two systems, where the MDO has an additional negative ligand bound to the iron center, and of course that withdraws electrons or donates electrons, and that changes the properties and the stability of this iron oxo complex. Probably this bond becomes more stronger, and it will bind more stronger as a result of that anion there. And we think that explains the larger stability of the iron 4 oxo complex in MDO with respect to CDO. So overall, this comparative studies of cysteine and mercaptopropionate dioxygenase systems show that large cluster models are quite useful for these studies. We have used them with more than 300 atoms, so that include the first and second coordination sphere. Mechanistically, they react very similar with consecutive oxygen atom transfer reactions with relatively small barriers. I think the largest one was around 10 or so. But both enzymes start from a open shell singlet and then through a spin crossover react on the quintet further. So there is a spin crossing that is important in both of them. But in both of them, we do see that the second oxygen atom transfer seems to be quite, could be rate determining in the reaction. Okay, so to summarize the non-heme iron work I discussed today is we did QM cluster as well as QMMM calculations on a range of non-heme iron enzymes, mostly dioxygenases. We've seen that these QM cluster models can give some very accurate representation of enzymatic or system. We've benchmarked rate constant against experiment, also spectroscopic parameters, which I didn't show, and the large model that incorporates the second coordination sphere particularly does well. So we calculated various catalytic cycles. and propose mechanisms, and we show especially that the second coordination sphere is important for substrate positioning, selectivity, and activity, but sometimes it's essential for the charge distributions and the local dipole moments and so on, to get a substrate activation on a position that you normally wouldn't expect, because that's a strong bond in the gas phase. Okay, so in principle I can stop here. Vaibhav needs to tell me where I am with the time.
Yes, we almost ran out of time. We have maybe couple
okay,
of...
then I stop here. I think that's best that I stop here, because otherwise I talk for another hour. I can just show my acknowledgement if that works. Yeah, I just show my acknowledgement because I need to thank a few people who helped me with the work. So most of the non-heme iron work I showed today was from two PhD students in the group. Hafiz Saqib Ali is he's from Pakistan, but he's now doing a postdoc in Edinburgh, and he graduated I think 6 months ago. Yan Ting is finalizing his PhD study. He's a PhD student from Taiwan. He works on the TEMP-A, and he's also working on herbicide biodegradation. George Ye was a master's student in my group, actually, who as a hobby did a bit of DFT calculations. And as I showed you, he got some quite good results on CDO versus MDO, and he was involved in a few more projects he's been working on. So I hope he'll find a PhD position somewhere, because he's quite talented. I showed you one slide with work from Dr. Dixit in Guwahati on the electron transfer. I didn't discuss the work with Kurt Faber on the biofuel system. I did show some work we did with Devish Kumar in Lucknow on the cysteine deoxygenase. And I think the other one, Jim Warwick, I also need to thank, because he did the electrostatic interactions in some of the project and analyzed the proteins and so on. And I need to thank some of the funders we had, like BBSRC, British Council. A long time ago we had cost funding and as well as Conosite funding. And I thank you for your attention.
Yes, thank you very much, Sam. We may take one question if there is any from the audience. Okay, I guess not. So let me ask it that in the systems where you saw that QMMM is not giving you the expected results, why do you potentially think where is it going wrong? Is it at the boundary between the MM and QM region, or is it the error cancellation more important in the DFT?
I don't know if there's a lot of error cancellation in the DFT. So one of the things we didn't try with the QMMM, so what we've seen with the cluster models is that cluster model C gave the correct energetics and selectivity, and that has, I think, 270 atoms or 370 atoms, I lost counting, because it has the histidine charged. So we should have used that as a QM region in the QMMM to see that QMMM can also do it. But we lost energy at that stage, and we tried already a huge number of models, as well as these two QMMM set of data. And as I said, we did 4 different pathways for each hydrogen atom abstraction. So it's a huge amount of data that we obtained. So we should have done it with that large cluster model as well, because I'm sure if you use the correct protonation states in your second coordination sphere, then it should be okay. So you should get the same results regardless if you use QMMM or DFT cluster. And we should just, the student was leaving, and we ran out of energy basically to finish it up. So yes, we should have done it with QMMM with cluster model C for completeness, which we didn't do. but it's something for the future. Maybe we can try it.
So that's all. Very good. Thank you so much for joining us.
Thank you for having me.
We have to stop here.
Yeah.
But definitely in the future, we would like to hear from you more, and we'll definitely call you and count upon you.
Yeah, sure, no problem. As I said, hopefully in the near future we can start traveling again, and hopefully because yes. I was supposed to come to Guwahati exactly 2 years ago, and that was cancelled. I think last time I was at your place was 3 years ago, I think. I think 3 years ago, or 4 years ago, maybe 2018. So yes, hopefully we can start traveling again when all this Corona business is finished. Yes. Because yes, it's easier to attend conferences face to face and talk to people face to face rather than through the computer.
Yes, thank you so much. And for the wider audience, it's a reminder that we have another session tomorrow starting at 10 a.m. sharp. So please do join us. Links will be shared through LinkedIn and Facebook and other medias. So we look forward to seeing all of you there. Thank you. Goodbye. Bye, Sam.
Thank you very much. Bye.